Good morning, everybody. Good morning, Jim. Love you too. Love you too. Nice to see you again today under clearer skies. Obviously, it's kind of crazy, isn't it? How just like sort of cleared off and is gone. Welcome back. I want to give you a few announcements, and it looks like Candace maybe wants to come up and give an announcement as well. Come on. That's all right. Invite yourself. Do you want to say something first? Okay, so I just have a few things to remind you of. We have posters today. Not all of them are up yet, I don't think, but I want you to make sure that you know they're over there. We are going to have a sort of a shotgun session a little bit later today, but make your way over during the breaks and lunch to have a look at the posters and get a little word from the folks that are representing them. So keep that in mind. We are also going to do a little something different today over the lunch hour. First, our development director, Quinn, is going to share a little bit with you, and particularly about something I didn't mention yesterday. This is why I'm terrible at it. There's a card in the middle of your table. We happen to be in the middle of a donation campaign because we have an anonymous donor with a spinal cord injury who committed up to $100,000 to match as a donation for our organization and for our work, because that person loves what we do and how we do it. So if you are inclined to donate or want to share that with folks that you think might be inclined to donate, there's a card on every table with a QR code. You can grab it, look it up, make a donation, or share it with your people. The other that I said yesterday also, but I just want to remind you again, please go out. There aren't many sponsor tables out there, but please go out and make sure you say hello to the folks from Inuvo and Kennedy Krieger and Shepherd Center and Emory. I'm missing somebody. Who else is out there? Onward, that's right. Our lone representative from Onward who made it. Please go out and talk to them and say hello. Ask any questions you might have. And the last thing is, maybe, do you want to share that? I think that's probably why you're here. Yes. Yeah.
Okay. That's good. All right. So then stay with me because make sure I don't lie. So good morning. My name is Candace Floyd. I'm a professor at Emory University. The Emory Entire Center is the one of the sponsors for this program, and we're really excited for you to come to Georgia and be working with you. And I'll talk a little bit about that center later today. So it's hard to see from these lights on stage, and I hope I'm not embarrassing her, but we have a really important member of our team, Laura Grabowski. She's a person with lived experience. She's also a fantastic scientist. And Laura and I work together with Unite to Fight folks to write an NIH R13 conference grant.
Yes.
And Laura and I have a great grant track record, by the way. We've written 2 grants together. We've gotten 2. So far, we're like rocking it. But and part of that conference grant, Laura had this fantastic idea that I want to talk to you about us to do at lunch today. And it was on this theme of, I keep saying voice, but it's perspectives,
Point of view,
different point of view. And so what we thought we would do was something, volunteer for this. This isn't something you have to do. And if no one does this, then fine, we'll just eat lunch. But the idea was that you would take 3 minutes. We'll bring the xylophone in here because I really want Ernesto to talk and I want to see if he can do it in 3 minutes. But so we'll bring the xylophone in here and we'll do the timer for 3 minutes, but just share your point of view. And so it's your elevator speech. It's something you want to tell us. It's your favorite dad joke. It's, it can be inspiring. It can be witty. It can be a thought you had. It can be anything. So just share with the group as a way for us to connect something from your point of view. Can answer the question why you're here. It could answer the question, what do you want to see? It could answer the question, why do you hate tropical storms? It could answer any question or ask any question that you'd like. Okay. So I think our plan is at lunch after
Quinn's going to come up, right?
Yeah. We're just going to come around with microphones, indicate in some way that you'd like to share a point of view. Like I said, we'll play the xylophone when your 3 minutes is up and let's see how it goes. Right?
Yeah, I'm just going to add a couple things to it.
You would.
Yeah. yesterday was a really lovely day, I think, and I hope I felt that in the room. I don't know if it's because we're a lot smaller than normal, because we lost a lot of folks this storm. The way the agenda unfolded, the way you all engaged with the content and the discussion, as you probably have guessed, for those who haven't been to our meeting, particularly scientists, we try to make this a very discursive meeting, more so than most scientific meetings. So there's a lot of chatter, there's a lot of talk, there's a lot of question and answer. We really try to do that and try to make sure we afford the time. This thing today is really an opportunity for you to just share anything. It might be reflections on yesterday, things that were discussed, things that maybe weren't discussed, things that you'd like to have heard discussed. And just give you just one little image or metaphor. I've used it a couple of times in presentations, mostly at scientific meetings when they're looking for a perspective from the SCI community. I've put up I don't have it with me, but I put up a slide basically that shows every stakeholder in this space: scientists, clinician, people with SCI, a funder, industry, regulators. They're all looking at yeah, well, that's kind of community, right? I mean, you could break it down way for much further, but the fundamental stakeholders all are looking at a particular target, a way they might define a win. And the key, and something I was trying to talk about a little bit yesterday, is those wins aren't always aligned. What is a win for a scientist may not be a win for us, or what is a win for industry might not be a win for us or a regulator. And sometimes they align, and sometimes a couple of them align. But the key is for all of them to align. The only way that we all win is if we all win. And so the idea of sharing your perspective is also sharing what is your target. And so people can hear that and understand that from the various points of view. It's a very important thing, and it's a kind of tuning in a sense of what we're all doing here and what we're looking at and what is a meaningful win. And part of that is everybody sharing their perspective. Okay? Good?
I have one more thing to say that I'm,
Okay.
So, remember that we have ombudsperson here.
Yeah.
Angela Dash is here. As you heard yesterday, she's in the back. She's wearing this most beautiful jacket today, so she can easily pick her out. She can be contacted through the app. She's in room 302. There are take-home snacks in room 302, just in case you decide you need a snack, and bottles of water and stuff over there too. So if you need to reach out to Angela, please do. She'll be in our point of view discussion as well this afternoon. And then again, this is supported by this NIH, NINDS grant that was in partial support of this symposium. And I want to thank the National Institute of Health and NINDS for that opportunity. Okay, now I've done my bit.
All right. So let's get into the next session. Mark, come on up. If you recall yesterday afternoon, in one of the videos from Onward, Gregoire said emphatically a couple of times that spinal cord stimulation isn't good enough. It's not a cure. It's not the thing, and it needs to be better. Well, that lends itself to this next session intentionally focused on combinatorial approaches, combinatorial strategies. Most everybody, I think, thinks or knows that combination approaches of stacking 2 or 3 or even more approaches, therapies together in combination are the way we are really going to have the greatest impact on spinal cord injury. So we've invited 3 presenters. First is Mark Anderson. Thanks for coming to talk about his work.
Thanks, Matt. Where's the clicker for the?
Clicker's right there.
Okay, great. This light is vicious.
Yeah.
Anyway, thanks for inviting me. It's my first time at this meeting, and it's been great to be here with all of you. And I'm really excited to show the research that I'm doing. And I want to be upfront with this. Everything that I'm showing is a biological proof of principle in a mouse. We all know from Dave and Jim's talk yesterday that even getting a simple thing as an external stimulator through the FDA is very difficult. So you can imagine if you have a regenerative therapy that's based on viral vectors, how challenging that would be. But I just think that if we don't start, we'll never get there. And so I hope that what I show you inspires you to dream about what's going to be possible in the future of spinal cord medicine. So my laboratory is embedded within NeuroRestore, which a few months ago, Jason and Matt likened to the Miami project to cure paralysis. And he's not wrong. So our collective focus at NeuroRestore is to bring together the best people in the world to solve what is arguably one of the most challenging aspects of modern medicine. So spinal cord injury was originally discovered by ancient Egyptians over 3,000 years ago. And while they didn't know what was going on the cellular level, they did recognize it as one of the most devastating things that can happen to a human being. It wasn't until about 120 years ago that the first neuroanatomists began to ask, well, why don't these nerve fibers regrow? And we've come a long way. We know that this lesion site is not one thing, but it's actually 3 cellularly distinct compartments comprising a fibrotic scar surrounded by an astrocyte border. And on either side of that, you've spared but actively remodeling neural tissue. And work that we've done in our lab has demonstrated that regeneration fails for 3 primary reasons. 1, you have an absence of neuronal growth capacity in neurons above the lesion. 2, you have a non-supportive environment within the core of the lesion. And 3, you have a lack of guidance molecules to facilitate growth of axons below the lesion. And our work has demonstrated that if you use gene therapy approaches to upregulate these growth programs above the injury, to remodel the lesion into producing supportive substrates, and to guide axons using another molecule, that you can overcome the failure of axons to regenerate. So the top image here is a control animal. The axons are in red, and you can see that they all stop. And if you apply this therapy, axons are able to not grow just into the lesion site, but completely beyond. And this is important because it showed for the first time that spinal cord injury regeneration is a very complicated problem. You don't need to manipulate just one thing or 2, but in fact, you need this combined and sequential reinstatement of 3 factors in order to get regeneration to occur. If you take one of those out, all this growth that you see goes away. So what's the point of this? So the point of regeneration is to restore function. So you can imagine how disappointed we all were when every single animal in this study looked like this. So despite this regeneration, this failed to restore walking in every animal. So this suggests that there are other mechanisms that were clearly missing that are required to restore function. So we thought about this. So following certain types of incomplete injury, you have natural and spontaneous rewiring and recovery of function. So the next slide that I'm going to show you, it's a very artificial injury model. It never occurs in a human. And it's important because it demonstrates that you're able to have regeneration, you're able to have rewiring and functional recovery in the direct absence of connections from the brain to the level of the spinal cord injury. So it's a double cut lesion. So you make one cut on one side of the cord, it paralyzes one leg. That leg recovers after a couple weeks. Next, if you separate this, you put another cut a couple weeks later at a different site, the animal loses the ability to walk. But over time, neurons from the brain synapse with those neurons that are between the 2 cuts and restore walking. It's not perfect, but it's a lot better than complete paralysis. So why are we looking at this? So we thought we can use this model because these neurons that are in between the 2 cuts are the neurons that we're targeting. They're called propriospinal neurons. And we thought that if we understood how this worked, we could combine this into our therapy to restore walking. So look at all those neurons that are between these 2 cuts. So there are actually, I think, 4 dozen different types of neurons in the spinal cord. So we wanted to know well, which one of these is important to mediate functional recovery in this model of injury that we saw? So the two questions we asked first were, well, one, what neuron populations in the thoracic spinal cord project to the lumbar spinal cord? And that's important because the lumbar spinal cord houses the walking centers. And two, which of those neuronal populations undergo the most transcriptional change following injury? So basically what we wanted to get at is how can we identify relevant aspects of this biology? And to do this, we've been developing single cell tools to enable us to answer these questions. Now, these are machine learning algorithms, and they're relatively complicated, but I think that you can summarize it pretty simply. So if you have two separate conditions, so you have uninjured, so you have a healthy mouse, and you have a mouse that underwent this incomplete spinal cord injury. What these algorithms do is it prioritizes neurons that you see on the left into, well, it ranks them. How much change have they undergone? And the idea is that if they undergo the most change, then it suggests, it doesn't prove, but it suggests that they're probably doing something that's important. So when we applied this method on these two conditions, it pointed out that one neuronal population came out as undergoing the most profound transcriptional change. And these are long projecting neurons that express a marker called VSX2. And so we looked at these, we traced them. This is an intersectional tracing strategy. And what's important is to show that these neurons, they're a specific neuronal population. They reside in the thoracic spinal cord, and they send projections directly to the walking centers in your lumbar spinal cord. Next, when we looked at them using immunohistochemistry, what I want you to take away from this slide is that they express a receptor for a growth factor called GDNF. So this means that one could use GDNF to target these specific cells. Third, we looked at, well, if these things reside in the thoracic spinal cord, what neurons from the brain send projections to them? So the VGI resides in the reticular formation and is a key walking center. And when we looked at projections from the VGI to these long projecting VSX neurons, we found that they synapse with them. So this demonstrates that they have all the key requirements to restore walking following injury. So we engineered our repair strategy and we targeted these neurons. So we overexpressed different molecules above the injury using gene therapy. We applied our injury at T13, and then we delivered different growth factors to turn on supportive substrate within the core of the injury. And lastly, we engineered a lentivirus to express GDNF, and we put gradients of it in the lumbar spinal cord. So this is a control animal. So you can see there's a complete absence of any neurons into or past the injury site. And when we applied this therapy, we found that not only did these neurons grow past the injury site, they actually went down and terminated in their natural target region of the lumbar spinal cord. Now, because we're scientists, we want to verify, well, what types of neurons is this therapy targeting? So we performed an experiment in which we sequenced these neurons. So we applied this gene therapy and we put a retrograde virus. So you put that below the injury site, it travels up the spinal cord, and you can dissect out that piece of tissue above the injury site, and you can sort it, fluorescently sort it, and sequence it. And when we looked at, well, what are we targeting? We found that we're actually targeting a number of different cells. It's not 100% specific, but we knew from the data that we're primarily targeting these long projecting VSX2 neurons. So what does this do functionally? So this is an animal that has not received a therapy, and this is 8 weeks after the gene therapy is produced. And you show that walking is not perfect, but this is the first time in my career, I've been doing this for 13 years, and this is the first time we've achieved weight-bearing recovery of walking in a model of anatomically complete SCI. So there are no neurons either going down the spinal cord or up into the brain. And this recovery is dependent on these specific neurons that we've regenerated. So this is an important study because it shows that not only do you need to get neurons to grow across the lesion, you need to get a specific type of neuron to grow to a specific space. And only when those requirements are met is recovery of walking able to be seen. And this is really cool, but I didn't tell you that one factor as simple as age can completely destroy this entire regeneration that you've seen. So this is a young animal that has the regenerative therapy. So it's a young adult animal. This is the same therapy applied to an old mouse. So this old mouse is maybe, it's hard to gauge this, I don't know, 50 or 60 years old. But you can see that a factor as simple as age needs to be considered when you're applying a regenerative therapy to a human, for instance. So this is the example of the walking. So on top is a young animal, the bottom is an old one. And so we wanted to say, okay, what in the world is going on in this old phenotype? And we wanted to understand that. So this compelled us to perform an atlas of a single cell atlas of spinal cord injury. So we did this, we sequenced over 500,000 cells and identified 94 populations. And when we were designing this experiment, we thought, well, we wanted to sequence every possible condition that we would think might be interesting. So we had uninjured, we had different time points, we had different severities, we had different models. So we had crush, we had a contusion, we had a hemisection. We looked at the comparisons between old mice and young mice, male mice and female mice, and different drugs that have been used throughout the years, like methylprednisolone, monocycline, chondroitinase ABC, and as a control, we used GFP. And what stood out as the most interesting was the difference between young and old mice. And to put that into perspective, the difference between the transcriptional difference between a young and old mouse is as large as the difference between an uninjured spinal cord and an injured spinal cord. And when we looked at what is going on, we found that neuroprotective border, so you have that astrocyte border, which walls off inflammation and prevents the damage of neural tissue. We found that these borders are completely malfunctional with age. And I'll show you an image here. This is a young mouse. This is spatial transcriptomics data. And the red is inflammatory cells. Same injury, an old mouse. So you can see the lesion is just completely exacerbated. Inflammatory cells have penetrated healthy neural tissue, destroyed neurons and axons in the surrounding area, and results in paralysis. So this is the white is an astrocyte marker called GFAP. The red is an inflammatory marker called CD45. This is a young mouse with what we call a moderately severe injury. And this is the same mouse, an old mouse with the same injury. And you can see that the lesion is essentially anatomically complete as far as we're concerned. This next thing shows the penetration of blood, which is contained within a young mouse, and the old mouse is like that. Functionally, the young mouse is at the top. Over 2 weeks, it recovers the ability to walk, while the old mouse, same injury, fails to recover that degree of function. So I've been working on astrocytes for a long time, and I knew that specific growth factors can stimulate their proliferation. So I went back, I collaborated with my friend who designs gene therapies, and I packaged these growth factors into a lentivirus. So these growth factors, EGF, FGF2, and VEGF, and we delivered them after a spinal cord injury in an old mouse. So this is an old mouse on the left. And following treatment, you can see that the formation of these neuroprotective borders is resolved. So what does that look like functionally? Sorry, one sec. So you can see at the bottom, you've restored the ability of an old mouse to walk more similar to a young mouse. And the reason this is really important is, classically people think of regeneration as like, okay, we're going to give something, we're not really going to think about what type of injury the person's had, what age it's at, is it acute, is it chronic, kind of where is it in time, because that's all going to influence the requirements to repair spinal cord injury. And why am I showing you this? We're not here to design cures for mice. They're useful as biological proof of principle, but our eventual goal is to put something into a human. And the only way to do that, we've talked about the limitations of mouse models, and I completely agree. You can only learn so much from a mouse. Regeneration therapies necessitate scaling whatever findings you have up into larger animal models, such as non-human primates. So I've spent the last 2 years developing a crush model of spinal cord injury in a monkey, and this is showing the deficit. And the reason I think that this is really important is because classically in non-human primate experiments, people use these lateral hemisection cuts. And the reason I don't like them is because the monkey walks fine after 2 weeks anyway. So if you're applying a regenerative therapy using a cut model, how much can you really learn from that? So if you invite me back next year, I hope to have more data on the non-human primate model. And with that, I'd like to thank everybody in NeuroRestore, and I'd like to give a special shout out to some of our funders, the End Paralysis Foundation, Wings for Life, and Bob Yant and his wife, Ann Kanowski, who are generous supporters of our future work. And thank you.
Morning, I'm Candace Floyd. I am a professor at Emory University and the director of the entire center, which I'm going to talk to you a little bit about today. In just a second. I am going to give you a little disclosure. So I consider this a little bit of a pitch deck. And so those of you in business might know what that means, right? And so this is pitching and discussing an idea today. So this is not more of one of my more traditional kind of scientific-based talks. And I think this is important because this idea came from this organization, really, and our collaboration with this organization. And it involves a lot of perspective and points of view from persons with lived experience. So I like to show this slide because this is very dear and near to my heart. And so we have a research environment where all voices and all perspectives are welcome and encouraged. My pronouns, I identify with the she, her, and hers pronouns, and I describe myself as a person with red hair and green eyes. And just in case you needed someone to add to your holiday shopping list, my clothing size is large. So as you see from this illustration, in our viewpoint, the viewpoint of persons with lived experience is critically important to the research that we do. It's incorporated in the research. And as I mentioned, this research idea that I'm describing to you is at the idea stage. And so we're really interested in hearing your thoughts. So I'm going to talk to you a little bit about this vision of the entire center. The Emory Neurotrauma Translational Research Center is a new organization that we've set up. A lot of the team members are here today, so you can talk to others about it. The other mandatory thing that you must do is you must take at least 1 to 10 piggy banks home with you so that you then remember the pigs as a good model. And also you make all of the children in your life very happy, or maybe use that to save up for your very next large endeavor. So what do we do at the entire center? So you've kind of heard about this valley of death already. You've heard about the bridge too far. You've heard some of these concepts. So the idea of the entire center is to produce a resource center that can bridge that valley of death, or can be a translational bridge between some of the preclinical research to the clinical research. And so our overarching premise then is that if we have these rigorous, reproducible, transparent, SOP-driven, good laboratory practice type experiments and improved injury models, then that will accelerate discovery and translation. And that we can use those resources in a collaborative way with the field and the community to help do that. And we kind of like, kind of say this in a quip way when I say that, pig models are what we are experts in. They're not easy to do. And just to give you an idea, you've kind of heard some things where they've said, for example, we heard yesterday people say, oh, it takes a lot of space, it takes a lot of resources, someone might have a few pigs. We have almost 60 pigs on census right now, right down the street, right? Many of them spinal cord injured, and half of my team could not be here today because we're still caring for some spinal cord injured pigs that didn't read the protocol and didn't make the time points like they were supposed to, and we have an extended schedule. So it is actually, you're correct, it's not a trivial endeavor to set up and run a really rigorous large animal spinal cord injury lab. I have an excellent team that does that here, and we have a lot of capacity. Okay, so this is just a picture of the team, at least the team at its last standing. I also just want to emphasize that part of the role of the entire center is not only our expertise in neurotrauma and pig models of neurotrauma, we also have, as we said, persons with lived experience on our team, but also a clinician network and a regulatory network. So the parts and pieces are in place then to work collaboratively with you all to take those therapies that are discovered in the rodents, use the pig as a translational intermediary, and go to the next step. So our main expertise, what I would say is kind of our niche and our thing that we can really bring to the team is this idea of reverse translation or back translation from the clinical arena to the pig. And the idea is that the discoveries that are made in these rodent models, like the fantastic ones we just observed, we have that network set up so that we can then use the pig models with clinically relevant outcomes. Those outcomes are set up, those outcomes are established in the pig and use that as a translational bridge. Okay, so why is the community support needed? Why is it really important for us to be here and us to be discussing this idea with you today? So the community support is needed to accelerate the research, as we've talked about, to direct research priorities, and also to support the validation of tools. So I had the honor yesterday of meeting with several persons that are from the clinical side. We met with several persons with lived experience, and they really have given us really interesting, innovative, and unique approaches to address this important question. So the project that I want to talk to you about today, look at that cute little AI generated pig, like adorable, right? So they don't look that colorful in the lab. But the project I wanted to talk to you about today is this idea we call epidural plus, epidural stim plus. So this concept, I would say, is something that we've had for a few years, and we'll talk a little bit about the timeline and the trajectory. But the concept is modestly straightforward at its roots, right, the brass tacks. So the idea is to take clinically relevant, clinically used epidural stimulators. So our goal here is not to make the best stimulator or to reinvent the wheel or to use, I don't know why that clicks and I'll go back, to reinvent the wheel. The idea here is to take things that are already being used in the way that they're being used in the human clinical trials and the clinical applications, and then add to that a combination therapy. We're a little bit agnostic here. Gene therapy is great. A drug therapy is great. We can do pharmacokinetics and pharmacodynamics in pigs. We can do neuroimage guided targeted gene therapy in spine, in brain. We can do instrumentation. We can do device. So this could be drug, device. In this example, we're going to talk about drug. So the idea would be, in this example, take an epidural stimulation protocol, add to that a plasticity enhancing, a novel plasticity enhancing drug that has come through these very complex networks of discovery that we've already talked about, and then evaluate those in the pig model using clinically relevant back translated outcomes. And as we spoke with a couple of you in the audience yesterday, sexual function, we talked about how we can do that, right, in a pig. We talked about how we can look at bowel and bladder management. This is more than you wanted to know, but there's a glitter that animals can eat, sparkly glitter that animals can eat that can give us transit times. right? So there are a lot of ways that we can evaluate these clinically relevant outcome measures in this model to address this question of combination therapies. So why use the pig? We saw some fantastic mouse data. I feel like an old mouse this morning, so there's that. Why we saw some primate. I really applaud that new primate model. I think that's a fantastic way to look at things. The pigs are also a really great model. And let me just give you a few advantages of using the pigs. So first off, we're more like pigs as humans. We're more like pigs than you would probably imagine. So pigs have been used successfully for decades in biomedicine. Their particular area of pride, I don't know if the pigs are proud, but a particular area of impact has been in coronary artery disease. So you may already know this, but pig vasculature is so similar to that of human vasculature. The heart is so similar that those components can be used in human transplants. It's similar with the skin. So the skin of pig is so similar, it can be used in burn victims or persons with burns for transplants, and skin grafts. Pigs are amazing in toxicology testing, infectious disease, and of course, really integral in vaccine development. Pigs are readily available. So unlike some other species that have more limited accessibility, pigs are readily available. As you know, they have we had bacon this morning for breakfast. They have a wide representation in both production, agricultural settings, and also in research settings. So there are genetically modified pigs. The genome has been mapped, proteomes have been mapped. There are maps of their brain. We're working on mapping their spinal cord along with others. There are pig models.
The generous donation that.
Sorry. There are pig models of lots of different diseases. So regarding injury, the anatomy of the pig spinal cord is very similar to that of the human spinal cord. The vertebral anatomy is similar. The brain anatomy is similar. They're gyrocephalic animals. They're socially complex animals that communicate with each other using bioacoustic signals that we can hear and interpret. So our laboratory speaks pig, right? So we can understand situations by the pig's communication with us that the pig rates as a distressful situation or a pleasurable situation. So these are very powerful tools. Oh, I went the wrong way. Okay. So
the generous donation that we received.
Let's talk a minute
Stand
about
Up to
this.
Cure Paralysis.
Oh, I'm sorry.
Is
That's probably from the slide. I'm gonna skip the slide. Let's talk for a minute about this project. So this project was generously funded by a consortium of foundations that Unite to Fight brought together to try to set up this platform. So the therapeutic drug that we are in conversations about testing in the first kind of testing iteration of this is the drug by Neurophysiological. They're here, I think. I think there's representatives here. And the idea that I talked to them a couple of weeks ago and they said, we're still really on board with a combination therapy strategy. We'd like to see more development of the epidural stimulator component in the pig first, and then we're interested in the combination strategy. So where are we in the development of this project? So we had this generous donation from these founders, and I think I'll go back to that slide in a minute. I think it's got some voiceover on the slide, which is causing us trouble. About 2 years ago, we have not started the experiments yet because we have been waiting on the receipt of the clinical stimulators. We have a generous donation of stimulators that were explanted from patients. The challenge with those explanted stimulators is that when the surgeons explanted them, there wasn't an intention to re-implant them, right? And so they're kind of in a bunch of pieces and parts. And so that's maybe not our best place to start. We have a generous donation from Abbott to receive some stimulators, but we are still waiting on those stimulators to be processed to the lab. So one way that you could help too is if you are a person that has readily access to stimulators, or even if you're good at repairing explanted stimulators, let me know because that would be the next step. So the research plan then is very similar to what you've heard in other plans, right? And so we evaluate sex as a biological variable. So we have both male and female gonad intact adult pigs. The idea then is that the pigs we'll use kind of 2 stages of research optimization. So the first stage of this optimization we call the platform optimization stage. And that is to kind of set up this profile where we conduct a contusion compression spinal cord injury in the pigs, both male and female pigs. implant the epidural stimulator in ways that it is implanted using human protocols that are reverse translated to the pigs. Using activity-based learning, so it's body weight supported treadmill training in the pigs with optimization protocols back translated from the clinic. We will then optimize that e-stim to that individual, right? And get as much out of that stim in that pig as possible. Once we've reached that functional plateau, then we will provide the combination therapy. So the dependent variables, as we talked about, locomotion. One of our novel aspects that we are developing is a way to establish and measure volitional movement in a pig. So as you will see when people talk to you about their experiences, often we saw many videos where the physician will say, move your limb in this way, or we'll see a video of a person with a stim on, performing something that's volitional. So in our hands, the pigs then have to press a pedal to get a treat. Pigs like treats, so that works pretty well. And then that enables us to really measure volitional movement. We have some back-translated methods to measure sensory sensation neuropathic pain that I'll discuss to you, spasticity, including human rating scales, bowel and bladder, and blood pressure, as well as sexual function. So the first step is platform optimization. Once the platform is optimized, we're anticipating doing that in the next few months. As soon as we can get a stim, we're ready to go. Kind of sitting on go, if you remember your Monopoly game. The next step then is using that optimized strategy to add a combination therapy. We're open for collaboration. Bring those therapies. Let's scale those therapies up and advance the science. So am I at 2 on my time? Yep, I don't know what my timer looks like, but okay. So I'll very quickly then just give you an example of a back-translated outcome that we use. And I want to talk for a minute about neuropathic pain. As was described yesterday, pain is not an area that is as readily studied in the research laboratories. So we use this method called quantitative sensory testing. Quantitative sensory testing is a method that has been developed internationally. There are champions of this in Germany. And so the German Research Network on Neuropathic Pain, the DFSN, is the paper that's cited there. That is the protocol that we have back translated to the pigs. So this DFSN protocol for measuring neuropathic pain in human populations is exactly what we use in the pigs. So I've actually been to Germany and been trained. As you can imagine, I was the only person in the room being trained how to do this in pigs, right? Everyone thought that was quite remarkable. But the idea is that we use all of these outcomes exactly the way they're used in human and the pigs, including the equipment. The QST, or quantitative sensory testing, looks at thermal detection thresholds, it looks at pain thresholds, it looks at mechanical detection thresholds. And I'm not going to go over the time or the methods that we use to do this, but we have evaluated an outcome measure in pigs where the pigs rate their pain on a scale of 0 to 10. And that includes vocalizations, as I mentioned to you, and other behavioral ethograms and behavioral outcomes. It's a very reliable and robust method. So here's some examples. So as you can imagine, after the spinal cord injury, we test the pigs using this quantitative sensory testing in different dermatomes, right, to get a dermatome-specific evaluation of the neuropathic pain profile. So as we learned yesterday, both in an artistic presentation and we heard in talks, there are varying ways that the neuropathic pain presents, right? And we're thinking about putting the X's on that body diagram as was shown to us in an artistic interpretation yesterday. So each pig presents with their own presentation of neuropathic pain. I'll show you a little bit of data on that. Different dermatomes are tested. In the human testing, the QST is typically done in one setting, lasting about an hour to an hour and a half. The pigs and the people testing the pigs, because remember, we're testing sometimes 6, 8, 10, 12 pigs a day, right? The pigs are in a body weight supported sling after spinal cord injury, so they're in anatomical approximation. And the team is working around these pigs to induce the quantitative sensory testing. So because of the team and the pigs' fatigue, we usually test 1 or maybe 2 outcomes per day across a week. And this goes repetitively over a semi-acute to chronic timeline after spinal cord injury. So here's a little bit of data from this, just kind of giving you an example of both region specificity and kind of some of these robust neuropathic pain responses we see after this mid-thoracic contusion compression injury. So what you see in the panel there of region 1 is the top bar that I don't think I have a pointer. If I do, I might break it. But that's around the 300-gram withdrawal threshold represents pigs that had a laminectomy only, right? So these are pigs that did not have a spinal cord injury. And then you see the spinal cord injury bar is the one denoted by the triangles there. And it takes some time, right? It's 5, 6 weeks, 8, 10 weeks sometimes in pigs after the injury that the pigs develop this hypersensitivity to mechanical detection. Region 2 has a different presentation than region 1. And again, that varies. The presentation of the termitomes that are affected varies from pig to pig. So using this type of analysis, we can use a pre-post design, right? So we can evaluate the animals prior to injury, induce the injury, and evaluate the injury effects of the animals. And then in this combination strategy, as we're describing, we could look at the effects with the stim on, look at the effects with the stim plus the combination strategy. So you see that this could be a very powerful tool. The pain threshold is a loss of function at the bottom there. This is just a little bit of data to highlight sex differences. And so we heard a little bit about aged mice already. We haven't heard a lot about sex differences, but I think there are likely to be pathobiological, biological effects of sex on outcomes after spinal cord injury. And it's something that our lab has been looking at for a very long time. And as I said, you know, we have specialized tools to have male, gonad intact male pigs in the lab with spinal cord injury because they require quite a bit more management. And so what you'll see on the side where it says cold testing is that there isn't a lot of cold pain in these pigs, in this pig model. And then on the right side where it says hot, you can see this very robust difference that starts very quickly after injury in differences in pigs responding to a warm thermal stimulus. But I want you to also pay attention to the pie graph there on the side. Males are represented in red and females are represented in black. And this is looking at the percent responders. And what you see is that 84% of the female pigs had a robust response to heat stimulation as a neuropathic pain trigger, where only 67% of the male pigs did. And so that is an interesting difference between the responders. And so between males and females. So as I reach the end of this, I just want you to think about this again. Like I said, it's not a heavily data talk, but more as a pitch talk for us to think about ways that we could collaborate. So the entire center is set up to collaborate with you in whatever way best serves the acceleration and development of new treatments. So you can either work with us and we can test therapies, we can come to you, we can combine together our knowledge and expertise to really accelerate treatments. I hope I've shown you that the porcine models of CNS injury are clinically relevant features because the pigs are very predictive of responses in human medicine. Pigs are readily available. Like I said, I've got a whole building full of pigs just a couple of blocks away. They're important tools in the future development, and we have a lot of these tools in place, so bring your ideas. So thank you again for all of the hard work, and thank you for the collective sponsorship of this combined program.
A couple quick things. One, as I said yesterday, we're having folks scribe the meeting. And I forgot to tell you all, when you get a microphone to ask a question, say who you are, where you're from, wherever you want to say you're from. Secondly, before we just jump into this, I want to jump on something Kajana just said, because I was thinking about it also during Mark's presentation. To understand where these are in the continuum of discovery and translation, right? So Kajana just said, we're a long ways away from understanding this translationally. Mark, if you caught earlier in his presentation, they delivered gene therapy before they injured the mouse, which is, don't be shocked, like that's how you do proof of concept studies. So what that means though, is that these are very early stage discovery work. And so I wanted to make sure that you all, particularly in the SCI community, understand where it is in the continuum so that you're not thinking, oh, Mark, when is that? When can I get that? Because it's not there yet, and it's complex, elegant as well. Candace is, on the other hand, is a little more ahead, as she already talked about. It's very translationally minded in a large animal study. So keep that in mind with your questions. And then also, if anybody wants to ask a question because some of what they just saw went over their head, don't hesitate to ask. Put it like I've done in the past, say, give me 3 sentences about what you just described as a highlight reel of the work. So let's go to questions. Maybe.
Good
Jim,
morning.
please.
Hey, Jim Hamer. I'm going to go off Candace's idea, 6 foot tall, white male, bald. Hazel eyes.
Beautiful, bold and
Um,
beautiful.
So thank you all for your presentations. I may be going down the obvious road. Mark, your presentation, first thing I started thinking about, because we've talked about this for years, are you going to go to larger models? You did that. Candace obviously threw out the opening for, so I'm just going to throw it out there that, like, Mark, your work, you know, we always talk about collaboration, what worked, what didn't work. Sponsors don't necessarily publish or researchers don't publish, but I think this is a great opportunity, Mark, for you to take your work with what you're doing and maybe collaborate with Candace and vice versa to get that larger model. Just throwing it out there, but you know, I know it's obvious, but I'm, you know, that's what we're here for. So hopefully it's a great option and we'll see something coming from it.
Comment.
Mouse, pigs, and monkeys is a winning formula.
Oh, the granting agencies would love it. So,
We have some questions from the live stream.
Please.
So specific to Candace, and I'm going to ask these in concert together because I think they're going to have a similar answer. So the first question is, instead of waiting on fixing those explanted stimulators to be repaired, could 2 to 4 stimulators be purchased and applied to get data sooner? And then similarly, they state that many small startup drug developers want to jump from mice and rats straight to humans, but the primary concern is cost of testing in these large animal models. So could you speak to the average cost per animal to conduct this research in pigs? And is it possible to lower that cost as well?
Okay. So the answer to number 1 is yes. And we're pursuing the purchase of these stimulators to get things started. If anybody has any, 2-for-1 special deals, let me know. But yes, is answer number 1. I'm going to answer the next 2 questions together. And I'm going to kind of do it in reverse. So then I'll give you some costs. So our lab spent, the leadership of our lab spent quite a bit of time in training for good laboratory practice. And for those of you that aren't familiar with that terminology, it's essentially documentation and quality assurance and the types of ways data is collected that is required for investigational new drug and device applications to the FDA. And so one way that we can reduce the pricing is we can do everything under near GLP to GLP. And a lot of times people wait on kind of that as later stage, and we can do that in a cost-efficient way. We can also do both safety and efficacy testing in those large animal models. And so I won't get into lots of details now, but we can do repetitive blood draws and CSF draws, and we can do pharmacokinetics and pharmacodynamics. And so the idea is that we can press some of those individualized steps into that one experiment. So essentially we get more data in a high quality way from that experiment. That's the only way to reduce the price. So what does 1 pig cost? So 1 chronic pig, and again, this is kind of ballparks or whatever. So for the lay members of the audience, we pay a per diem, so each day the pig is in the vivarium, we pay for that. In rodents, it's probably what, $1 to $2 a day?
Mhm.
Yeah, it's $30 a day for a pig, just to kind of give you the idea, right? It's much more expensive. So essentially 1 pig experiment and a chronic experiment is about $10,000. Including pig and personnel costs. That varies. And we do have very active worksheets to help people do that. But yeah, there is some cost involved for sure.
Can you also just tack on the difference in duration between an acute pig study and a chronic pig study, and what's chronic in a pig?
Yeah, it's not a answer that anyone had. So this question, you guys probably can chime in on this too. So this question between acute and chronic and how timelines translate between rodents and pigs and monkeys and people and what the biomarkers are for acute to chronic is not just an answer that I have, right? This is an answer that the field is working actively on. And how chronic is defined, often is defined in plateaus of function or stability of lesion or things like that, right? And so it's not this is me saying it's not a well agreed upon timestamp, but for us, chronicity in a pig looks to be about 3 to 4 months after injury. And so for our experimentation that we've described today, that's what we're targeting as a relatively stable lesion with spinal shock, immune responses are back down. I mean, they're never completely down. And that's kind of where we are with that. Some people say it must be 6 months to be chronic. Some people say it must be a year to be chronic. So the jury is still out on exactly what chronic means, but that's where we are. Is that fair?
Yeah, I think also it depends. I think you said it very accurately that it's also the plateau of functions, and it also depends for functional studies in the chronic phase, but general translation of what time in a rodent, which is what I'm familiar with, translates to human is I think that's kind of sort of right now agreed upon. 1 hour translates to about 4 to 5 hours in patients for acute studies for neuroprotective strategies. So just as a general translation. But again, for the chronic phase, I think in the rodents, 12 weeks is considered. And that's one of the reasons we've chose 12 weeks as anything between 6 and 12 weeks.
I'm going to make one more comment, which you guys can talk about, and I promise I'll be quiet. So pigs don't regain function naturally as much as rodents do. So I always kind of joke that rodents are made to take a hit and keep on ticking or whatever, but pigs do not have as much spontaneous recovery.
Right.
And so that may affect those timelines and plateaus.
Yeah. And just so that's clear to everybody, for like the caring for the animals up to that period, whether it's 30 days or whether it's 4 months, is before you do the intervention.
Correct.
Right. So there might be training right ahead of that, training, whatever the model is for getting a pellet out of a thing or doing the treadmill or whatever the training might be. But that's 4 months or 30 days of caring for an injured animal and then doing your, as we mentioned yesterday, your pattern manipulation, right? To feed back to pattern recognition. Is it Cynthia?
Hi, Cynthia Templeton from New Jersey. Thank you all for what you're doing. I have a question about whether any of you are currently using AI in your research and if you're planning on or how you think that will change and help everything that you're doing.
Yeah, I mean, we use it in our studies of single cell sequencing. So you have all of this data. For instance, the algorithm that we showed uses artificial intelligence to rank the cell types. So it definitely aids in finding different potential targets.
Not in the study that we presented here, but in the lab, we also have some work looking at decoding the brain signals in response to various interventions. So we're working with data scientists and using AI to decode that signal using AI as well.
We have a neat collaboration. So thank you for the question. Our collaborator is Bob Data from Harvard University, who uses AI to evaluate what he calls cognitive ethograms of behavior. So it's kind of cool. You know, the pig is in this big arena and all these cameras are set up and they use AI to evaluate subtle changes in the pig behavior and interpret those in a functional recovery setting. And so instead of us, we typically will use like a behavioral ethogram, right? And we predefine what behaviors we're looking for, as I described in that pain research. And in this AI collaboration with Bob Data, we take an open approach to discover basically what the behaviors of the animal are telling us using complex AI. So we're excited about that new collaboration.
Trayvon.
Hello, my name is Trayvon Dixon from Indiana. My question for the gentleman with the regeneration study. With regard to regeneration, hold on, I'm trying to see what notes I read. I'm glaring on my screen. Have you considered STIs and that are non-traumatic in nature and how they might impact your research for like STIs that don't have a break at all, for instance, IFTM? which is an autoimmune disorder where my immune system attacked my spinal cord. And MRI that I had 6 months post-injury has my spine is completely normal, but I lack function, obviously. So just how that would impact your study in any way?
The short answer to that is no. We have not looked at any of the non-traumatic spinal cord injuries such as that. I'm not honestly really that familiar with animal models of them and even the pathobiology of them. I'm not really sure how one would tackle that. I'd have to look into more of that. I don't have a better answer, unfortunately, than that right now.
I may just add, we use not an immune attacking model, but I do have a model in which we do a non-traumatic progressive compression spinal cord injury. And I think that is a big gap in the field. Not that, of course, we're focused on traumatic spinal cord injury, but there's a lot that we can learn from, as you described, from the non-traumatic injury models that we can subsequently apply. So one of the mechanisms that we discovered for the interneurons was initially using the non-traumatic. So because there are lacking a lot of good models of non-traumatic injuries, that we don't know a lot of the pathophysiology of that diseases. So I think
I would say for transverse myelitis that that is an emerging area of research. And I think a lot of the research, particularly with immune modulation, some of these restorative strategies that are in traumatic injury are likely to be also very applicable to transverse myelitis. So I don't think it's a situation where it's not considered, right? I think it's considered in the larger viewpoint of spinal cord injury, inflammation, regeneration and repair.
Do you think that's a consensus across the field that strategies for traumatic would be applicable to some of the non-traumatic?
Yeah.
Do you think that's sort of a shared view? Yes?
I think so. I mean, there are investigators that, for example, use ALS, as well as there are people who use strategies that are worked for breathing in traumatic spinal cord injury to ALS and vice versa. Just knowledge can be translated as well as potential treatments.
Hi, my name is Rayhan Qureshi. I'm a student at Georgia Tech here today with the Translational Neurotrauma Center at Emory. This is a question for Dr. Anderson. I was wondering, I thought it was interesting how you mentioned the astrocyte barrier as kind of stopping the neuronal growth down the spinal cord. And so I was wondering, pertaining to your methods with the injection of the neurotrophic factors, if that was a treatment that you had to continue through multiple iterations or if it was a one-time thing, because my concern is that maybe in more chronic time points, I know yours was 8 weeks, in more chronic time points, maybe if that astrocyte barrier is not deteriorated, if the astrocyte barrier is maintained in the spinal cord, if that could end up in more chronic time points reversing what you did with the neurotrophic factors, essentially, just reversing the treatment.
Sure. I mean, to answer your question, the manipulation of the astrocytes and the lesion core is a once-off thing. When you look at the growth that's been achieved in the studies, it grows through, it remains at chronic time points. That being said, if that same strategy is done chronically, and by chronically, the definition is arbitrary. What I can tell you is that if you do that treatment 1 month post-injury, it doesn't work. And my guess on that, I mean, even when I was doing the surgeries for these, you try to inject a 1-month-old fibrotic core of a mouse, I broke the needle a couple times. I mean, the density of this fibrotic core is really high. And my assumption is that that's what's blocking and preventing the growth. And fixing that will likely require other strategies. I know Jen Doolan's working on cell grafts. That's probably likely going to be a requirement for repairing more chronic injuries. And a lot of the stuff we really just don't know the answer to yet. do. If anything, it makes it stronger.
One more question, then we're going to go to break.
Yep, got a question for Candace. So I'm Brian Canavan. I'm with the Neurotech Institute out of Columbus, Ohio. We're a nonprofit aimed at bridging academia and industry to get approved medical solutions for neurological conditions, mainly spinal cord injuries. My question's at a bigger scale. One of the things that I believe keeps these discoveries from being translated to industry and ultimately people in collaborations is IP ownership. So I'd love to hear the setup, if you've cracked that, with collaborations and ultimately the IP of these discoveries as they go forward. Are you a vendor for fee, or is the IP then get caught up in the dreaded tech commercialization office of an academia?
Yeah, it's a great question. And I will say that we continuously, so cracking things is one of my superpowers because I'm really persistent. So we continually crack it. And the answer is all of the above. And so we have a couple of relationships right now, some of them that were developed at this conference with small biotech startups, right, where it's critical they maintain their IP. And so together we cracked, the office is enough to make sure that that was done. We are hired fee for service. We do that all the time. So it's almost like a little mini CRO kind of, and that works really well. When the company comes in, they've got IP. We work with Emory's IP office and we say, we are here to run the experiments. Near GLP, we've got the model, we've got the expertise, and we will, give the results. So that's one strategy. In situations, we have a company that we're working with now that I think they think they're a different stage than Candace thinks they're at, right? And so there's a lot of development in their drug administration and all these things that need to be worked out. And in that case, I'm like, you guys, we're going to spend a lot of time developing devices and administration strategies for you. Yeah, Emory's going to want some of that IP. So it just varies based on where the group is in the spectrum. And I would say our team has the spirit, which I hope I've conveyed today, that I don't know if I can cuss here, but you fill in the word. We want to get through the BS to accelerate the therapy. So these are some of the most respected folks in the field, and we keep picking on nature papers in this conference. And so, these are nature paper folks. We're not the nature paper folks. We're the translate that folks, and you take it if you want it on. We don't, once you've done that, you don't have to talk to, So that's kind of the way we see things, and we work through Emory and with the companies to develop an IP strategy that's effective. But it's a good question.
Okay, thank you all. Appreciate it. All right, everyone, let's gather back. We're going to get started into our next session. And I just want to give a quick pause to reflect for those who were here yesterday, particularly in the session in the morning, which, maybe some felt like Kent apologized multiple times for bringing everyone down and just taking a machete and beheading everyone. Sorry for the imagery. Challenging these questions about the system and about that pipeline of from basic research and discovery all the way through to clinical application. And so here we are in this next session. And I want you to even think about, if you can remember some of the things that were discussed yesterday that Kent brought up about some of the obstacles in translation, how the money is spent, and then from David Baker, which I hope you all caught, because I know I had to shut him off because he was over time, but essentially the slide you might have missed, and I just want to quick say it, is he was basically saying academic science isn't the place you should look for delivering what is industry or commercially ready. And basically what he said is we need an alternative in academia, and suggested that you put that kind of like translational institute, translational incubator, which some academic systems have, embed it in the university in a particular area so that you can ready things to come out. And that was the thrust of his discussion. But in light of all that, we have 2 presentations and then some bad news that the third one will not be here and will not make it for actually the second time, because she couldn't get her visa from the embassy, which is really unfortunate. Chinese national living in London or Leeds will not be here. So we'll have a little extra time. After having said all of that, I would like to introduce Daniel Mikol, Chief Science Officer, correct? CMO, yes. Who's going to talk about nerve gen and provide a little background. And then following that will be Mikol Lauw from Lineage Cell. Please welcome Daniel Mikol.
Okay, good morning, everyone. So as you can probably guess from the title, I'm going to be talking about clinical translation as it relates to spinal cord injury. And is there a problem? And I would say that there is, because to date, there really hasn't been successful translation in spinal cord injury. Treatments that have shown efficacy in animal models have not shown efficacy in humans. So that's a failure of clinical translation. And clinical translation isn't easy in any therapeutic area. I've worked in my industry career in a number of different indications within neurology. And it is fair to say that in other therapeutic areas, in other areas of nervous system disorders, the success rate is better. So I think there's got to be some opportunity. And I'm thinking about all of this from a clinical perspective, but really the bridge from animals to humans. So in the presentation, there's really 2 main parts. Number one, I want to take a look at how translation has been approached in spinal cord injury to date, and then think about how we might perhaps change things or do things differently to make things a little bit more successful. So what I'm showing you is the path for development of any therapeutic, beginning with preclinical studies of efficacy and safety, onto phase 1, 2, 3 studies, and hopefully an eventual approval. The clinical translation refers to the bridging of efficacy from the animal studies to the first studies in humans with the condition of interest in what's called usually a phase 2 proof of concept study. And the goal of this kind of a study is to determine if there's an efficacy signal. And I use quotes around that because that can mean different things. But the main thing that we're looking for is the therapeutic doing what it's supposed to be doing? And I think, unfortunately, there have been some negative trials, and we don't know the answer to that question. We haven't learned a lot. And the phase 2 proof of concept trial is really an excellent opportunity to learn for the first time about the effects of a treatment in humans. So I want you to think of this trial as an obstacle. It's an obstacle that you have to overcome in order to get further on in development. And if you can't, then perhaps that therapeutic is going to die. And so what I'm saying about phase 2 being an obstacle in development is reflected by this slide, which shows probability of success. And success means getting all the way from either phase 1, phase 2, or phase 3 to an approval. And if you're in phase 1 or phase 2, the probability is lower. And we're looking at 7 different therapeutic areas, and nervous system disorders are the white bar, by the way, which is the lowest among the 7 bars. But once you can get past phase 2, things look a bit rosier. and the probabilities increase quite a bit. So again, the phase 2 proof of concept study is an obstacle. And if you can demonstrate that the drug is doing what it's supposed to be doing, you've learned something and you can build on that. There are many reasons for potential failure of clinical translation. I want to boil this down into really a simple model. And so think of a preclinical study as simply consisting of an animal model, a treatment, and some corresponding placebo, and then outcome measures. And what you're trying to demonstrate is that your treatment improves outcome measures compared to vehicle or placebo. It's pretty much the same in a clinical trial. You have a population that you select, and this is defined by eligibility criteria in the trial. You have a treatment, which is the same as in the preclinical study, although there may be some differences in dosing, et cetera. And then you have outcome measures that you've selected. So in this simple model that I'm showing you, where things can go wrong and failure to translate is, first of all, if the wrong study population is selected for the clinical trial, it doesn't match the animal model. And think about it. The humans with spinal cord injury, it's a very heterogeneous population. Everyone knows that. There's so many variables. There's the level of injury, the severity of injury, the type of injury, recovery from injury, et cetera, et cetera. It's not that way in the animals. I mean, you have the single strain of animals, and there are strain differences in terms of how they react to an injury, a spinal cord injury. You have a single strain, and they pretty much have the same type of injury and severity of injury. So we're introducing a lot of variability just by going into humans. And that's for really any type of animal. And the second thing where things can go wrong in this simple model is if the wrong outcome measures are selected. And by that, I really mean you want to have outcome measures that are sensitive enough to detect a treatment effect. It'd be very unfortunate if you have an effective therapy, but you can't demonstrate it because you don't have the right outcome measures in the trial. So let's take a look at how clinical translation has been approached to date in spinal cord injury. And the next slide shows, it looks like a complicated table, but it really isn't. I'm going to break it down for you. The left half simply refers to some preclinical studies for 8 different therapeutics on each of the 8 rows. The right half relates to the clinical trials that followed. And the second column, I don't have a pointer, but it refers to the type of animal model. There's a lot of abbreviations there that aren't important. What's important is that for each of these 8 interventions that I'm showing you that was tested in preclinical studies, they demonstrated improvements, improved function, and therefore went on to clinical trials. So as we look to the right half of the slide, you can see that most of these clinical trials are completed, not all. All but one are placebo-controlled, and for the most part, they're acute intervention studies because in the animal models, that's where efficacy was demonstrated with acute intervention. Now, the 2 right columns, I want to spend some more time talking about. First, the trial population, and then the primary efficacy measure. And I'm going to introduce the Asia Impairment Scale, and this isn't really introducing to probably the majority of the audience, but I'm going to assume that there may be some of you out there in the audience or online who aren't familiar with this, so I want to go over it just briefly, because this has been used to define the trial populations of pretty much all the trials to date. It's a simple scale of impairment after spinal cord injury. And really, the impairments are Asia Impairment Scale A, B, C, and D. E is normal. A and B are more severe and motor complete, which means there's no residual motor function below the level of the injury, whereas C and D are incomplete. So I'm highlighting complete red and incomplete yellow. And then I'm applying it to that column. And you can see that the types of individuals who enrolled in these studies are mostly Asia Impairment Scale A and B, motor complete. far fewer subjects enrolled were C and D. Why am I spending so much time talking about this? It's because the animal models, in the models that I'm showing you here, these animals have residual function below, motor function below the level of the injury. They're motor incomplete. So I'm highlighting them all yellow. And you can see this is a disconnect. And so when I came to NerveGen 3.5 years ago, it was my first experience working in spinal cord injury. I looked at this and I just couldn't understand the disconnect. And I think it is a potential reason for failure. It does introduce a significant risk because the people that are being studied in clinical trials, they don't really match what was being evaluated in animals in the preclinical studies. Now I'm going to talk about the primary efficacy measure. And as you can see, the most commonly used primary efficacy measure is related to upper extremity motor score. Many studies use total motor score or upper extremity motor score, abbreviated here as UEMS. And so I want to talk a little bit more about this, and I'm going to again assume that not everyone in the audience understands what this means. It's derived from what's called the INSCI examination. What INSCI stands for is shown at the top left. This is a form that's filled out really for every spinal cord injury patient. And it's a way to document performance on a motor examination as well as a sensory examination, how that may change over time during recovery after injury and following rehabilitation. What's relevant to calculating motor scores, I've highlighted in yellow on this slide. And really, it's quite simple. 5 muscle groups in the upper extremities, 5 in the lower extremities on both sides of the body are given a grade of strength from 0 to 5, where 0 is no strength, 5 is completely normal strength, 1, 2, 3, and 4 are in between. You add this up and you can get a maximum score of 50 in the upper extremities and 50 in the lower extremities. This is very useful in clinical practice, and that's why it was developed. But it was never developed to be used in clinical trials. That's how it's being developed, and as I showed you, it is by far the most common primary efficacy measure in clinical trials. And coming from the outside 3.5 years ago, I still don't know the answer why. I think part of it is because it's so useful in clinical practice, it's trying to be translated into clinical trials. I have no idea how regulators view this because there's no real sharing of information from some of these failed trials. But coming back to the motor score, this wasn't developed to be used as an outcome measure in clinical trials. It is somewhat subjective, and if you really drill down, it's ultimately based for each muscle group on an ordinal scale from 0 to 5. It's not a linear scale. Going from 0 to 1 or 2 to 3 or 4 to 5, it's not the same, but it counts as the same. And moreover, motor recovery isn't stepwise. It's a continuum. But with the motor score, we're artificially making it a stepwise process. So the bottom line, again, kind of as an outsider coming into working in spinal cord injury, I think the motor score may very likely be insensitive to change, to demonstrating change, unless you have a very effective treatment. And to put that another way, I think a treatment may deliver a clinically meaningful effect and not show a change on the motor score. So I think we have to keep that in mind when selecting outcome measures for clinical trials, especially primary outcome measures. And before I transition to really the second part of my talk, I want to leave you with one additional insight from 3 recently completed trials of riluzole, the no-go receptor decoy, and anti-no-go monoclonal antibody. For each of these trials, the primary outcome measure was based on motor score, and for the overall populations in each of these trials, the result was negative. But for each of the trials, subgroup analyses done after the trials were completed suggest a potential benefit in the subgroup with incomplete injury. And I'm showing you the results for riluzole on the right, which has been published. It's also been published for Axer 204. And I've seen the results for the anti-NOGO antibody just at ISCOS last week, and that will soon be published. What does this mean? Well, I think it might suggest that residual connections may be necessary for the best possible recovery, which doesn't seem that crazy. But this doesn't mean that if a treatment is eventually shown to be effective in an incomplete population, that it won't be effective in a complete population. What it means is that if we are striving to determine if a treatment is doing what it's supposed to be doing in a proof of concept trial, it might behoove us in this initial trial to enroll individuals with incomplete injury to give ourselves the best, highest probability of demonstrating a beneficial treatment effect. So let's come back to talking about an efficacy signal, which is really the objective of a proof of concept trial. And in such a trial, you're going to include various clinical outcome measures, but it's an opportunity to include what I'll call a surrogate efficacy measure, a biomarker. And Bob Temple, who used to be at the FDA, defined a surrogate efficacy measure as a substitute for a clinical outcome measure where changes induced on this biomarker are expected to predict changes on a clinical outcome measure. Now, it can't just be some random biomarker. There should be a link between the biomarker and the clinical outcome measure. And ideally, the biomarker is upstream so that the clinical outcome measure is actually dependent on what you're measuring in the surrogate measure. And I'm going to give an example on the next slide to make this more clear. But because biomarkers can be more sensitive to change, this can enable smaller and more efficient trials and can increase the probability of detecting an efficacy signal in a proof of concept trial. So here's just a general example of a surrogate measure in cancer, solid tumors. Let's say you have a treatment that is designed to be effective in the treatment of people who have solid tumors. Ultimately, in a clinical trial, you're going to want to demonstrate that you can extend survival in patients who received the treatment compared to placebo, but that's going to require a lot of patients and a pretty long study duration. That's what is done in a pivotal trial, phase 3. In a proof of concept trial, you can conduct a smaller trial, a shorter trial, and demonstrate that your treatment reduces the tumor size. I mean, this should seem intuitive. If you can reduce tumor size, you may have a beneficial effect down the road on survival. This is what has been used successfully in cancer trials, solid tumor trials, where change in tumor size is the surrogate efficacy measure. Well, what about spinal cord injury? We don't have an established surrogate marker of efficacy today, but I just want to think about this kind of out of the box. It's fair to say that clinical trials to date have focused on evaluating whether a treatment improves motor function. And that's been mostly tested by looking at the motor score, as we just saw. So maybe one way to think about surrogate efficacy measure in spinal cord injury is as follows. Many conditions are progressive, like cancers, they grow. Spinal cord injury is a singular event, traumatic spinal cord injury. It happens and there can be recovery after that. So maybe, whoops, wrong way. What we can do is during the spontaneous recovery period after spinal cord injury, try to identify biomarkers that actually predict better or worse motor outcome. And then we could apply that in an investigational trial to predict motor recovery. Again, as a surrogate efficacy measure in a proof of concept trial. And I will propose that one such surrogate efficacy measure is electrophysiology and motor evoked potentials in particular, which is a measure of descending connectivity. With spinal cord injury, there's a loss of connectivity, and electrophysiology measures connectivity. that provides objective quantitative information. So I'm not going to go into the technical details about motor evoked potentials, but they give you information about the magnitude of the electrical signal that gets all the way from the motor cortex down to a specific muscle group of interest and how long it takes to get there. But what's important is that improvements in motor evoked potentials have been shown to predict improvements in motor function, both in animal models of spinal cord injury and in humans during spontaneous recovery after injury. And I'm going to give you an example now of how we are applying electrophysiology in our proof of concept trial with NVG-291. And in the next few slides, I'm not going to go through all the details of this trial. It's more about how we're approaching clinical translation. And so again, we have this, we're trying to bridge results from preclinical studies to studies in humans in a first proof of concept trial. And so when we were thinking about how to design this proof of concept trial with NVG-291, we started looking at what we knew was important from preclinical studies. And the first point is that efficacy was demonstrated in both acute and chronic intervention models by several different independent investigators. And so we wanted to explore this in people because we really don't know if NVG-291 is effective, if it may be effective in with acute intervention or chronic intervention or both. So in the trial, we are enrolling two separate cohorts of people with subacute injury and chronic injury. It's essentially two trials in one. The second point from preclinical studies is we've already talked about is that the models that were used are motor incomplete models. And that's why we decided early on to enroll individuals with incomplete injury in this proof of concept trial. And third, the preclinical studies showed that NVG-291 could improve functional recovery, motor recovery, bladder recovery. And it did so by promoting axonal regeneration, plasticity, as well as remyelination. It enhanced connectivity. And so in the trial, we want to measure connectivity. And we're doing this in a variety of ways, but the principal way is by measuring motor evoked potentials. So to sum this up, in this trial, we're enrolling two separate cohorts of chronic and subacute motor incomplete cervical spinal cord injury, defined chronic and subacute as shown on the slide. And we're not just including clinical outcome measures, we're also including electrophysiological outcome measures. Some of the key endpoints are shown on this slide. I'm not intending to go through the details. I just want to highlight a couple of things. One is our primary objective is to look at the change in corticospinal connectivity to two specific muscle groups, one in the hand, one in the leg. But we're also looking at 5 different performance tests in this study, mostly focusing on the hand and upper extremity, but we're also looking at change in 10-meter walk test speed. I think this is important because these tests are largely based on continuous scales. And with a continuous scale, for example, how much time it takes to complete a specific task, it can give you more information. As there may be recovery, it gives you more information than ordinal or categorical scales. And so it can increase the statistical power to detect a treatment effect on motor recovery. So I'm going to close now, and I'm going to do this in a visual way. First of all, remind you that the phase 2 proof of concept trial, really we should think of as a hurdle in development, and we need to think about it differently than how later clinical trials are designed. The objective is mainly to show that your drug is doing what it's supposed to be doing and you see some efficacy signal. You're going to include clinical outcome measures, but it's not necessary that you show statistically significant change on clinical measures in this first trial. Ideally, you're going to include surrogate efficacy outcome measures, as I showed you, can be very sensitive to detecting change, more sensitive than clinical outcome measures. Having done that, you can then focus on convincingly demonstrating clinical efficacy in subsequent trials. This is what's typically done outside of spinal cord injury. But in spinal cord injury, that's not the case. And you also have to be careful about selecting the right population and your list of outcome measures. In spinal cord injury, for the most part, efficacy surrogates are not being used. I think this needs a lot of development. It's not like there exists some obvious efficacy surrogate measures out there. I think that we have to think about what makes the most sense. And it may vary for a given therapeutic and what you're trying to demonstrate. But what you want to do in selecting an efficacy measure is you want to measure what you think your treatment is going to improve. Okay. So I think there is opportunity in trials of spinal cord injury, and we should consider including them biomarkers in studies, electrophysiology, but there's other potential biomarkers that can be considered as well, such as wearable devices. I think we should also give careful consideration about the clinical outcome measures that are incorporated into study designs in a proof of concept trial and try to include more functional performance tests, again, because they're based on continuous scales that can be more informative in an early study of a therapeutic. And the last thing I want to say is there is the potential with a surrogate efficacy measure for it to be directly translatable. You can't do that with a functional measure because, you know, animals are built differently than humans. But for example, the motor evoked potential is directly translatable. You can measure it in animals, you can measure it in people. And so if you study it in animals first, in animal models of spinal cord injury, and you show that you have a beneficial effect, and you carry that over into humans, that can, to some extent, decrease the risk in clinical translation. So I think this is something else that we should consider. And that's it.
Thank you, Daniel. I anticipate folks are going to have a lot of questions for you. And which we, as I mentioned earlier, Jessica Kwok's not with us. We'll have a little more allotted time for questions, assuming there will be lots of them. Next, we have Mikol Lauw from Lineage Cell. And those of you who follow Lineage Cell, are you familiar with Lineage Cell? You know, it was, and it was, and it was, all the way back to Jaron. Thank you, Jaron, from the beginning. Welcome, Mikol. Mikol was unable to, he tried very hard to get here. Thank you. Waiting on flights and trying to get here on his way back from ISCAS, I imagine, right?
A little bit of both. I have family in the Netherlands, so I was there for both. I was taking a detour through ISCAS and coming back, and Washington was very nice to me, but the weather in between me and Atlanta was not. So I'm very sorry not to have met you. I hope I can't see myself, which is fantastic, which I hope you're not seeing a humongous head, because that always frightens me when people dial in. So if you are, apologies ahead of time. And hopefully you won't see a dog dive bomb this presentation. So
welcome. Go right ahead. Thanks, Mikol.
Thank you for the invite, Matthew. So I already, are you going to start the presentation and am I just not seeing it?
I think you have to share your screen, Mikol.
Oh, I have to share mine.
Yes,
okay.
You're in control right now.
I'm in complete control. Oh, that's not a good thing.
Be careful.
All right, stand by. I have to, all right, hold on 1 second, sorry. Let's see. Now I have to, Zoom has to, I have to pick the right one because otherwise you know how that goes. Hold on 1 second. Okay, I have way too many things open. Give me 1 second. Sorry, didn't realize I was going to be the one to do this. So, Let's see. All right. I think I got the right one. All right. Well, thank you again, everybody, for taking time to listen to me and for inviting me to comment. I am sorry I wasn't able to be there in person. Can you just confirm for me that you're seeing the full screen on your sides? If someone can on the.
Okay, so if you could do me a favor and yep, up there you go. Perfect. Thank you.
Yep, that was the question. All right, great. So when I was putting this presentation together, I was looking at all the fantastic talks that were happening before and a lot of people who were talking through the science, and I was trying to think of how can I take our experience in the development seat for a product that actually is in development, and how can I help with the final question, which is your breakout this afternoon, which is how can the SCI community help in terms of product development? I am a social worker by trade, so my brain immediately went to, well, then instead of me spending a lot of time talking about the specific science behind the product we're developing or how that actually happens, I thought I'd spend a little bit more time on talking about who it is that influences the kinds of questions we ask. And when I say questions, I mean the research that we conduct, both in the preclinical arena, which Dan just did a great job of describing to you how that leads into the clinical arena, which is by which we mean doing studies in humans, and then even into when we're working with people who have injury and including them in our clinical trials, how those stakeholders continue to drive really what happens in a particular program. So that's going to be the clinical development of OPC1, which I'm going to talk to you about, is going to take a back seat and be the background for the discussion today. So I will say really quickly up front, Lineage is a public company, so I am required to tell you that some of the things that we will be talking about today are forward-looking statements, and certainly as such should not be taken as any promises of what may or may not happen into the future. If you're interested in anything about Lineage, you can certainly go to our website for our public statements. I am a full-time employee of Lineage Cell Therapeutics. That being said, any opinions that may arise during the question and or answer session during this are completely my own. So I couldn't see obviously in the room who was aware of who Lineage is, and who I thought we'd give a few seconds to just talk about who we are. Lineage is an organization that is spread across a couple of countries. You can see here our corporate headquarters are in Carlsbad, California, while our manufacturing occurs in Jerusalem in Israel. We have about 75 employees total across both organizations. Our primary core strength is in the production and development of cell therapies. So specifically, we look at, we have processes in place to ensure that we not only have cells that can be used in multiple different settings, but we look at the control and safety and reproducibility of those cells. So we have scalability. We have strengths in looking at the purity and identifying so that the actual cells that we're producing are the cells that we mean to have. We look at the potency of those cells, both ensuring that we've enhanced them to the extent that they are the most useful if they're transplanted into a patient, and as well that we can test that functionality before we even do that, and then ultimately that we're able to produce that at a scale that is actually useful and producible, and helps us to give access should we eventually have a cell that that's actually been approved by the FDA. And that a good majority of that then occurs at our center in Israel, as previously discussed. So our particular type of cell production is allogeneic, which means it's not necessarily coming from, it is not coming from the person who's actually going to receive it. It's coming from a registered cell line, which we, from pluripotent cell lines in particular, which are those cells that can turn into any cell within your body. We've mastered our ability to manufacture that in large scale, and our proprietary our technology, our intellectual property, is taking those particular cells and then differentiating them to what we like to call an optimized state, where they're going to be accepted by anyone without rejection, and that they actually look like the evolutionary determined cells that are in your body and can truly be a form of transplant, in the same way that a heart transplant or a kidney transplant or anything else can work. It can go, if in our particular case, there shouldn't be a difference between whether it's you, me, or someone I don't know. From that same cell bank, we can produce something that should be accessible to everyone. Our treatments are meant to be one time, and the beauty of being able to take that from a general cell line as opposed to taking it from each individual is it's much faster to actually produce those cells, and obviously to scale those up and make them available to everyone else. This comes into play later when we talk a little bit about the stakeholders. So just a quick, I won't go through this, but in addition to what we're looking at OPC1, which is our cell line that is looking to impact spinal cord injury, we have a few others down the pipeline, including Oprogen, which is for geographic atrophy, which is currently under research with Genentech, and then a few others coming up along the pipeline. So, drug development from a stakeholder perspective. So I want to take a quick moment to sort of set the stage and think about this first and foremost from what drives the questions that we're asking, both from how we think about posing those questions and what we're really looking to establish, and then talk a little bit in buckets of those people who are interested in those questions and talk about that and serve as the forefront of what the rest of this conversation is about. So first and foremost, when we enter a development program, we really think of this in terms of risk and benefit. And I think you've heard just in your discussion from Dan a lot about how we work about translation and look at that from can we prove that it does what it does? But when we think about a product, what we're also thinking about is what is the benefit relative to the risk that you're incurring, either in how it's being administered to you, what the product actually does to you in addition to what it's supposed to do, what's your experience while you're taking the product if it's over a long period of time. And while this sort of unintuitive little balance scale where you have the larger piece being balanced by a smaller one, you want to think about that when you think about drug development, because there's no standard grading scale for what safety means to you, what you are willing to risk that is more valuable than the benefit you might get from that. Who gets to decide that? On what basis products get approved, on whether you decide to do a particular treatment or not, that's going to vary. So if, for example, I have a treatment that can extend your life by a year, but there is a really good chance that that's going to cause blindness and permanent deafness in you during that time that you're still alive, is that worth it to you? Is that risk that comes with it worth the benefit of the extra year? Some of you may go, of course, I know exactly what I'm going to do, and other people may not. The quality of the life that you accept, the quality of function that you have are equally as important in developing the drug as is the actual benefit that it precedes. So keep that in the back of your mind as well as we think about who we can influence as we talk about getting to a commercial product. So when we think about considering a new potential treatment, obviously at the center of that is a person who's going to get that treatment. And we think about if we think about an industry from what is the need? What are the things that haven't been covered by medications a million times over? Or if they have been a million times over, what is the benefit that we can bring differently with our technology? We want to think about the safety. And when we think about safety here is very specifically what happens to you physically, or mentally, or in some way biologically impacts you to have, when you take this treatment, whatever it is, cell therapy, large molecule, small molecule drugs, what happens to you what happens to you and what do you experience, or what are the likelihood that you're going to experience certain things. We want to think about efficacy, which Dan's already done a great job talking about, so I won't spend a lot of time. Does it work? Does it actually do what we intended to do? And does it give you what you need to do? And then we want to talk about a few things that not as many people think about, which is tolerability. So let's say the safety seems okay. You're not going to die from it, but what it entails, you taking the drug, how you get there, the safety that you get is, let's just say, constant itching every day for the rest of your life. Is that still the drug you're going to take? The itching's not horrible. It's not really causing people to scratch so much they bleed, but it is constantly there. For those of you who've ever had a bug bite, I think you can attest to the fact that you want to get rid of that. Is that worth it to you relative to the efficacy that you're seeing? And then equally important to that is, who is that information collected from? And we as an industry are incredibly good at collecting that information from your physicians, from external data, from looking at observing things and saying, oh, this is how we interpret it. But what we haven't always been as good at is getting that data from the actual person who's experiencing that. And so that's a critical piece that is built into all development programs at this point. And finally, while it is always the bugaboo that nobody wants to talk about, it's what's the cost? What's the cost to develop it? What's the return on investment is what the industry side is going to ask you from. But on your end, is it, once you determine that cost, is that something I can afford? Who's going to pay for that? Who's going to cover that being there? And how can I get access to that particular drug? And so those are all sort of things to consider when we think about a new potential treatment once it enters into the industry arena, sort of moving past what Matthew was talking about in the intro to this of what happens at academics. This is now in an industry, in industry and biotech and pharma, how we kind of think about new treatments that come up as potential for full development. And so when we think about that in terms of who then are the development stakeholders, I put them into buckets. And if we're starting with the person, I'm putting that person, you, me, and everyone else into the side as being the center and focus of why we're doing this in the first place. And now I'm really looking at who drives where this development program goes in addition to that, and the information they take that we learn from you as you're exposed to the drug, we learn from you how that exposure, what occurred during that exposure, what actually happened to you, who is interested in that information and what do they do with it. And if we make industry the big bucket, the next thing down is the physicians. Now I could have grouped them with patients, and I would like to think that physicians are always aligned with what their patients would like. But I think, as we all know, physicians are looking at it from a different perspective. They're looking at you as the individual, but they're not you. They're making decisions based on their education, on their understanding of different medications, on the balance of things that they know across multiple patients. But what they don't know is everything that comes from you. So they will have potentially a different opinion on what should be accessible, what's appropriate for a particular person, what is the right risk-benefit balance. So it's a good thing to remember. Then you have the FDA. And the FDA's responsibility here is from more of a national and societal one. So what do we as a society, in our case for the FDA, what do we in the United States consider to be the appropriate type of drugs that we will look at approving? What do we as a society consider the right balance between something that should be approved and available and allow people that level of choice versus those other ones that we consider, no, it's not demonstrated safe enough. So whether or not that's impactful on one or two people, as a general rule, we don't want this accessible to the larger population. And obviously that's a balance they get from the data they receive from patients, from advocacy groups, from physicians, from long-term data that they've collected over the years of all different kinds of medications. It is also obviously something that is both objective and subjective. And to the extent that they don't have information, that they don't know what the perception is of patients and what their willingness is to accept certain risk-benefit ratios, that obviously compromises their ability to make those decisions on what drugs should be available. Again, keep that in mind. And then finally, again, to the cost question, there's the payers and the people who are actually going to provide the resources to make this drug available to people who need it. And they're looking at this from a cost benefit, both looking at it from safety in the sense that if we do this, is that going to cost more for this patient because of what will happen when they get this medication? Is how it's delivered going to be more expensive versus the actual benefit that they get? Is that worth it to us? Is giving and allocating that money to this particular medication appropriate? Because do we then lose the balance for other medications that might have a different effect or a similar effect that we think is good enough? And obviously another area that is both objective and subjective, objective being the components of what the additional care needs are, what the side effects are, what the other treatments are, what the components of that drug, what the effectiveness of that drug will be. And the subjective being, is that meaningful enough to us to differentiate where and who and what treatments we give? This is an overly complicated slide as I looked at it this morning, but it was meant to, in some respects, look at, to point out a couple things. One, everyone looks a little bit at the same questions. Whoops. We'll look at the same questions. They'll look at whether it's safe, it's tolerable, it's efficacious, what other options out there and cost and reimbursement. But each of these people I just talked about will look at this slightly differently. From physicians to payers to regulators, they're going to look at it from the patient's well-being. In the case of physicians, we'll look at it primarily from a cost perspective, and from regulators, what's the benefit overall and or the harm to society. And they'll look at the exact same data and interpret it from their lens. And from our side, both from the biotech and the developers, as well as the investors who help to support the development of those, we have to look at every one of those people's input and say, now, is this something that we're going to be able to develop? Are we going to be able to meet all the people's needs while keeping in mind at the center the person who's receiving this drug, so that the access to this drug, when we're done, if we're able to show it does what it does in a manner that is safe enough and on balance, something that people are interested in taking, can we produce this to and make it accessible to the people for whom it's intended? All right, so we're going to now take a little bit of a tour through what has happened with OPC1, and keep in mind all the things I just talked about in terms of who the stakeholders are, and hopefully I'll be able to frame some of the questions that we've asked through the different stages we've gone through development, and that will set you up for a little bit of success this afternoon to start thinking about and talking about where could you possibly, as a community, have impact as development progresses for any product. So before I do that, probably a little bit of science in here. I'm not going to spend a lot of time on this, but to talk to you about where OPC1 came from. So OPC1 came out of the research and thought about what oligodendrocyte progenitor cells, which are precursors to myelinating cells of the central nervous system, if we were able to create those and actually transplant them where they have been injured or and damaged due to the spinal cord injury, what could we potentially achieve? Knowing that these progenitor cells create myelinating cells, which means those are the ones that help insulate your nerve axons, so those things that help you help the neuron communicate and actually improve that particular process. Could that, in fact, because it's essential for the proper functioning of those neurons, could that, in fact, restore or augment the functional activity if you've had a spinal cord injury? And research in animal models would say that there's a high likelihood that there's a possibility for that. To Dan's point, a lot of that work has been done in incomplete injury, but nevertheless, there was a lot of evidence that built to say that that was a possibility. So OPC1 was a product that was then built for a cell transplant approach, where once it's been differentiated to the OPC1 from the pluripotent stem cell, it would actually take the place of those progenitor cells that you would normally otherwise have, and or support the ones that are still in somewhat existence, and then replace the function that was previously lost. OPC1 specifically is from an NIH registered cell line. As I mentioned before, these are allogeneic, so taken from someone else and then given to another person. And then it's meant to be a one-time surgically administered injection into the spinal cord. So OPC1, as Matthew mentioned, started at a different company many, many years ago. So there were Geron and Asterias were responsible for bringing this from its infancy into the clinical trial state. And there was a lot of technology that and background that had to be in place for that to occur. But once it was, they were able to establish all of the initial protocols to produce OPC1 and do proof of concept in animals, which Dan talked to you about a little bit earlier. And not specific to OPC1, but it's just in general what that meant. And they were able to open the first IND for any pluripotent stem cell derived any pluripotent stem cell that was derived from a line, which was the first time that happened in the United States. So obviously they were in its infancy. They were looking at things for the first time and working through that with regulatory authorities, with investors to help them to understand how this might work in the space that they were going into. They designed the first in-human study in thoracic subjects that were Asia A in nature, and there were 5 patients in that first study. And then that was followed when the product, because Geron had some changes in its business priorities, it was followed when Asterias took the product over from Geron with the cervical injury study, which enrolled patients in Asia A with Asia A and Asia B impairment. So lineage came into play in about 2019 when we looked at when Asterias was looking at how it was going to continue to fund this with a great interest in OPC1 for a variety of reasons. One, as I mentioned to you earlier, our strength is in development of different cell lines. So we thought we could bring some improvements to how that product was being manufactured. We thought about the need to develop the device that had actually been used in the previous studies and whether that was really scalable into the future and could we make improvements there. And more importantly, we thought about the fact that we needed better and prioritize that engagement with the spinal cord injury community, especially in consideration of how we actually measure the outcomes and the effectiveness of this particular product, looking both at the outcomes of the studies that had been conducted and whether when we move into the future, some of that needed to be optimized and changed and looked at slightly differently. We made sure that we also we also heard the message that people want to hear what happens to these trials, whether they're positive, when they're negative. And so in 2022, published the first 2 outcomes of the first 2 studies about 10 years into the one and 5 years into the other. And we'll talk about that a little bit later. So going back into the history, when OPC1 was first manufactured, obviously manufacturing, even in a development program, goes through different stages. And what you're trying to do is optimize it such that it is most scalable, that it is repeatable and reproducible, and that it is safe. And in the first generation, it was a fairly complicated process. It was a specialized process that required a great deal of time and expertise to actually produce. You had to do it centrally and then give it to the site, and then a very limited stability. But it was exactly where we wanted to be in terms of the type of characterized composition of the cells that were contained in the product. And so it was safe enough to move into people, at least from a manufacturing perspective. And then we looked at it from a preclinical perspective. So what do we need to know when we're looking at the mechanism, the administration and manufacturing, the safety and efficacy? What did we need to know to be able to enable a trial? And again, we think about that in this particular case, with heavy emphasis on what's the FDA going to need for you to demonstrate that you have the appropriate controls over your manufacturing, the right assumptions about using the proper models to think about what safety you might be able to predict that may happen in humans. Obviously, that's very different when you get to a true human model. And some evidence that there is a reason to believe that your product as it is actually going to have some level of efficacy, because if we're going to put people at risk, there should be some reason to do so. In normal is the wrong word, in clinical trials where the drug is in some ways safer and easier to administer, you might start in healthy volunteers. And I think a lot of you have probably heard of healthy volunteer trials. In a product like this, where it is installed surgically, typically speaking, or like an oncology trial where they're very toxic drugs, you typically would start in people who actually have in this case, injury, and in other cases, the disease. So the importance of having translational models for safety was really high. So we did a lot. Geron and Asterius across themselves did about 35 in vivo studies, and that may not seem like a high number, but it is quite a lot. And there were a lot of information that was obtained from that, including that the cells would survive, that they would go where they were supposed to go, and whether or not they would not go where they aren't supposed to go, that they're going to have the right, they're going to, in the places that they're going to have the right activity that we expected them to have. And to the extent that we were able to detect, do they do things that we hadn't anticipated? And did we observe that? And what meaning might that potentially have for people, in particular, when we consider the magnificence that is the human immune system, and whether there would be a reaction to the cells that they received? And overall, at the end, the conclusion to those 35 trials or 35, excuse me, in vivo studies, so animal studies, was that this was something that can, at least with risk, but safely move forward into humans. We found out a number of things about the mechanism. We found out that what we thought it would do, it could do. So we looked at this from perspective of preventing cavitation, which are the little holes that you have that occur when you have a spinal cord injury, and that it seemed to help fill those. It did create myelination of axons that were still in existence, as seen below here in the mouse model to the left. And then it had it excreted neurotrophic factors, which are those things that support the growth and survival of neurons, which are obviously important for messages to travel up and down the spinal cord. And we recognized that from the studies done in animals, that there were no additional neurological deficits, because that's obviously a concern if we're going into a spinal cord injury population. There was no allodynia, or at least increases in allodynia, where there's a feeling of pain from something that normally shouldn't cause you pain. And we saw a very low incidence of ectopic or tissues where there occurred where it wasn't supposed to occur. And the system with which we actually were delivering was also demonstrated to be something that appeared to be safe, at least in the animal models, or useful. And ultimately, we also saw in the animal models that there was lower locomotor improvement, so giving us some hope that this would be something that could actually be beneficial to people. And so we went into people, and I'm not going to go through this slide in any real effect, except to show you that even a simple trial where there's a single event of dosing, you need to consider what you're doing for follow-up for over a period of time. This sort of describes to you that our primary goal for these first-in-human studies is safety, as it always should be in a phase 1 study, which precedes the phase 2 and efficacy studies. And then we have the secondary assessments, which were the neurological functioning. For both of these studies, we looked at that from a primary and we looked at that over 15 years and found that we saw the same thing we saw in animals, that we had evidence that there was durability in the engraftment, which meant that we saw evidence of there being actually taking hold where it was supposed to. And in about 96% of those, in those patients, we saw no sign of lesions still at the cavity at about 24 months. And overall, it was safe to deliver to patients. We didn't see any neurological decline, no enlarging masses, no spinal cord injury, additional damage. And in the cervical study, which was done in more patients, we also saw that 96% of patients achieved at least one motor level of improvement. And in cohort dose 2, that we had some patients who even saw a greater degree of improvement. What that means needs to be looked at in larger studies, but at least, again, it gave us a confidence that there was a reason to move forward. Just as a quick note, these patients are all that we asked all these patients to be followed up for 15 years. Very normal for cell and gene therapies to look at 5, 10, and 15-year follow-up, or at least out that far, as this was really new at the time it was started. 15 years was the long-term follow-up, and here you'll see in 2026 and 2032, we'll have some outcomes to see how those patients fared. So I'm not going to go into a large detail into this because I'm running myself out of time, but to highlight a little bit where Lineage is taking this. So we looked very specifically at the mechanism by which we were delivering drug and saw some opportunity, again, to enhance the safety. The old syringe positioning device was very cumbersome. It was really huge. It was hard to manipulate. The injections were performed on a table mount. And more importantly, because of the movement of the patient, you needed to stop respiration to deliver the drug, which obviously, while nothing happened to any of the patients that was in any way permanent for the ones that participated in our studies, not ideal in the future. Sorry, Matthew, are you trying to reach me as I'm talking?
Yes, I'm hoping you can start to wrap it up, Michael.
I will. I got your text.
Thank you.
Yeah. So that study is on its way to improve to a new system which diminishes the overall risk, and that is a study that we're working on implementing at the moment. We also improved manufacturing, which is obviously our skill set, looking to improve the purity in actually the cells that we're delivering to make that, while we didn't again see any major risk from that, to hopefully make that more safe. And more importantly, we also are turning it into a thaw and inject formulation. So taking out the complexity of developing that and moving that into a formula that could be very easily transmitted to any hospital. So what's next? Bring this back to the conclusion of this particular discussion is, once we've identified the need, we're needing to look through providing the solution. So we're looking, as we look at what we need to do in this program, we need to think about that from the various stakeholder perspectives. So what's the clinical benefit? What's the benefit and how do we best measure it? I think Dan talked about that quite a bit. And I think what we're thinking about as well is, going back to the example of risk versus benefit, we need to consider what it is that's actually meaningful to you. So focusing on all of the other things listed on here, one of the biggest challenges is not only what change do we see and detect by our scales, but is that something that is actually meaningful to the person on balance of what it means to take this drug? What does it mean in combination with other drugs? Is this with other drugs, with other treatments? Is this something that when the clinical benefit comes out that actually has functional changes that actually impacts your quality of life in some particular way? And how do we best measure that and show that to help improve, at the end of the day, the ability to access these kinds of products? And that's it. Thank you very much. Sorry for going over.
Thank you, Michael. You can stop your screen share so we can see your head fill the screen.
All right.
For the Q&A. And Daniel, if you would. There you are. Come on back up. So I want to set the stage a little bit because I anticipate I'm sort of feeling the room right now. And there's two things I want to say, and then we'll get into questions. The first one is, I think a lot of you in the room, some of you have already come up to me and said, well, what about some details? So Neurogen is conducting a clinical trial as we speak, or is in the process of. Mikol, Lineage Cell has some plans. I'm sure folks in the room would like to know as much as possible as you can disclose at this point. So that's one thing. And then the second thing is, because I've also received a couple of texts. Michael, you put up and I'm in no way, I hope this doesn't sound like I'm throwing you under the bus. You put up a screen, because I'm not, of stakeholders. And it's something related to what I mentioned earlier this morning, that you'll notice the stakeholders in the earlier part of Michael's presentation were essentially customers, the customers that a company like Lineage has to go through to get to people with spinal cord injuries. And that means investors, that means clinicians, that means regulators, that means insurance companies, who's going to pay for this treatment. Those are all customers that sort of have to say okay before people with SCI get the treatment. It's a hard reality, but I wanted to point it out because, again, it's why we're having the conversation we're having and why it's important for every stakeholder to be at the table and also recognize that every target is different for every player. And how do we understand that? How do we navigate that? And in some ways, it gets to the conversation we want to have later this afternoon about how does the community help instead of just sitting, waiting or watching or waiting for any information to trickle out. Is there anything that we can do? So with that, I open up to questions. Okay, right here. A lot of hands came up. Barry?
Thanks, Matt. Barry Monroe from U2FP Praxis. This one's for you, Michael, but I think Matt just sort of set it up perfectly. Maybe you can just speak briefly about your latest energy, and I know Lineage and yourself and some of us in the room are involved in what is called a patient-focused drug development program with the FDA. Maybe explain what that process looks like, and we'll carry that conversation later on today, if you could.
Oh, goodness gracious, that's a couple of hours conversation in and of itself. So the PFDD is an FDA initiative to ensure that they try to engage the patient community to really understand what drugs are of value to them. And the first one was developed in about 2012, and there unfortunately haven't been as many as you'd need to. But in particular, in areas where they don't have a lot of expertise, where there's not a lot of understanding or patient experience data that I was talking about earlier, it's a process by which a patient-led group can help to engage the FDA to help bring that experience to them so that they understand when they're looking at the different drugs that are being brought to them, what should we be looking for in terms of drugs that are meaningful to patients? What are the experiences that they have so they can approve and or consider drugs in that context? So at the moment, there is a group that's working to put that together for the spinal cord injury community, or with the spinal cord injury community is the correct way to say that. And it will be led by people who have lived experience. And then there will be those of us in the background that are working to assist to build those engagements, to help look at raising funds, because obviously money is unfortunately, again, a component of that, of putting that together and bringing that to the FDA to and begin those discussions. And for those of you who are in the future ever asked to participate, so valuable to get different people's experience and to understand people, because each of you are the master of your own experience. I think no one person can speak for everyone or a group as much as we all like to. Certainly a big area that several different companies, as well as patients with lived experience, are working on to develop. Did that answer your question, Barry?
Yes.
It's really odd to talk into a screen and not see you guys. So
from the chat, I assume.
What's
Yes,
that?
I have some questions from the live stream. So I'm going to try and combine these. So for the Neurogen study, have you considered the error when using more than one study site to measure MEPs? And then as far as GRASP goes, as much training as there is for each of those study sites, have you considered using videotaping the tests to examine potential human error and maybe visualize what may be responsible for the speed of each of those tasks?
My mic on?
Yes, okay.
So I may ask you to rephrase that. So one question was about GRASP and
one was about MEPs and one was about GRASP. MEPs, if you're doing a single site, what about error? And in GRASP, have you considered videotaping to make sure as a control?
So first about the MEPs in the single site, and the reason we're doing a single center, it's not normally something you would do, but with electrophysiology, we really wanted to leverage it to get as much information as possible about this trial. As I spoke to briefly, it's much easier to do at a single center. So you're actually decreasing variability by using a single site that has experiencing in it and conducting these kinds of measurements. You have a small number of assessors. They have the same training, same type of equipment. There are different types of equipment at different centers. You don't have to worry about that variability. So really, for all subjects in the study, the advantage of a single center is you can reduce variability substantially with electrophysiological measurements, which is the primary objective of the study. So it actually helps us. It doesn't hurt us. And then as far as the grasp, I guess all I can say there is this is a secondary outcome measure that we're using. And we use trained individuals, and we aren't using video to evaluate it and then check, simply because, this is a proof of concept trial, and we're going to learn from it. But, I have other ideas for a next trial about what kinds of measures we might use, and I'm not sure, we may or may not use the grasp, but to answer the question, we're simply using people who are trained in the conduction of the grasp test, and we're collecting results.
I think just for a little context, and I hope I'm not saying too much because I'm not going to name anything or anybody, but the context of that question, I think, is in the variability of the tester. And so there's some certain concerns about, even in one center, if you have, say, 4 therapists, 4 OTs, right? And how they conduct and execute the grasp study varied in some studies in the past. And so I think that's probably the context of the question, as a cautionary tale to consider.
No, there's much we could say about the grasp. I think I like performance tests, and just to be brief about the grasp, there are really 4 subtests to the grasp, and performance test is just a part of it. There's 6 specific tasks that are measured, but there's also a motor evaluation, a sensory examination, and sort of a qualitative prehension examination. And I'm not sure if the question relates to the qualitative or quantitative prehension testing. And I guess it doesn't matter, but for my interest in this measure, more in the quantitative prehension testing, I think that could be useful. I think the potential downside of the grasp is it's a composite, it's arbitrarily weighted, and you're including things like motor examination results, sensory examination results, qualitative prehension, quantitative prehension. It's kind of all lumped together in a score. If I were to pick one, it would probably be the quantitative prehension that is the most important. Yeah.
And real quick, does everyone in the room know what the grasp test is? No? Did I hear a no back there somewhere? Okay. Can you just quickly explain it?
Yeah, I don't know if I can recall what the actual acronym stands for, but it's a graded test. You're basically measuring hand function. And like I said, there's 4 specific tests. You examine strength of muscles of the hand, sensation in the hand. You ask the subject in the study to perform certain prehension tasks, like pinching the index finger and thumb together. Just their ability to do that, to make sort of a cylinder with the fingers and thumb. And you get a score for qualitative prehension. We're using that to actually, for part of the eligibility criteria, both for minimum and maximum performance. And then the quantitative prehension is based on ability to perform certain tasks, like turning a key, for example, and that's being scored, and it's objective. But people who are conducting this definitely have to be trained, and that training is provided.
Great. Thank you. Candace.
Yeah, so I know
And
that
then,
vaccine development and therapeutic development are different. I understand that. But I guess my question for both of you is, what did we learn from COVID?
That's a very broad question and a provocative one. What were you specifically referring to when you say learned from COVID in terms of
get-
how to conduct trials or in
I
terms
guess,
of?
yeah, I mean, from the development, therapeutic development perspective. So I mean, we all went through that together to a certain degree. And the timeline was truncated, the pathways were abbreviated. I mean, so I guess that's kind of what I'm asking is, did we learn something that can be used to optimize and accelerate therapeutic development for spinal cord injury?
So I think I can start, Michael. I mean,
Yeah.
first of all, vaccine development is it's pretty standard, right? I mean, all you're doing is eliciting an immune response. I mean, there's a lot involved, but it's pretty low risk and you can accelerate timelines and there's precedence for it. It's been done before for so many vaccines and it can be accelerated. I think the point is around how can we possibly accelerate? And certainly, the entire world came together about trying to accelerate these vaccines for COVID. I think for spinal cord injury, we need one success first, a therapy to go all the way through development successfully, and then we can try to dissect the whole process and try to figure out how we can speed things up along the way for future products. But I should also say that I think there is opportunity for acceleration in spinal cord injury if we have evidence of a treatment working. Regulatory agencies are going to support this. I mean, there's going to be regulatory flexibility, just like there has been for other conditions that are rare conditions and have a huge unmet need. And advocacy is huge. So advocacy can help along the way when a treatment gets to that point. So definitely we should think about accelerating, but we kind of have to, I think, wait for a first success and then follow on it.
I think for me, the challenge in spinal cord injury isn't necessarily one of finding an accelerated pathway, but really identifying how it is that we measure success. And I think if you look at a number of the failure trials and the traditional way that a clinical trial is set up is you have one key primary endpoint. If you fail on that, knowing that spinal cord injury is really, the outcomes of spinal cord injury are really a multitude of factors, you're not technically allowed to test on those second and third ones if you do it in a hierarchical manner. So I think where the learnings really come from for helping us to accelerate some of that is understanding how we best analyze those and looking at other areas where they've been successful in grouping using co-primary endpoints, using a global statistical approach of taking multiple potential factors of success and having that count towards your actual detection of efficacy so that we can move past the, well, you didn't make this, but hey, you made all these other improvements, but you can't file that and move forward with that. And I think to the point of efficacy and to the point of the PFDD we talked about earlier, those are areas where the FDA has shown willingness to move, but that varies a little bit from division to the division. So bringing that to them and helping them to understand that this in particular area it's a multifactorial outcome, and therefore each product might have different outcomes in different people, and that they need to think about that and be willing to accept those endpoints in a slightly less traditional way than they might have, I think are equally as important. For me, the pandemic, what it brought was it pushed forward technologies that allowed for examining and observing what people do in their home space in a way that we weren't able to before, really making use and being able to translate Fitbits and iPhones and Wi-Fi for detecting movement and all those kinds of things And to the extent that those can be used in spinal cord injury trials to measure what the actual outcome is of people with respect to even motor function or day-to-day function, I think there's advantages to the volume of data we can produce to, again, show the multifactorial nature and of any product we put into play. So I think it was very valuable, but in and of itself, it isn't the pathways that were used for vaccines were not necessarily additionally useful in and of themselves.
Louise?
Matthew. Thanks to both of you, Dan and Michael. So I'm Louise Phipps-Semt from Blink of an Eye, and we have rapid response team that goes bedside to SCI families and ICUs across the country. And our navigators help the families in multitudes of ways, but we also are really like the translators for some of the SCI trials, because we've got this incredible close proximity to families with immediate buy-in, because all of our navigators are spinal cord injured people with lived experience. So my question for both of you is about the family stakeholder perspective. I do see that it is oftentimes left out of the equation because our loved ones whom we are advocating for in multitudes of ways, it oftentimes and usually requires their families to get them to the trials, to get them into whatever it is that's happening in the most cutting edge forms of science, from not only transportation and living, but caregiving as well. So that's the premise. The question might be more pointedly for you, Dan, with our excitement about the NerveGen trial. Have you given thought to the acute cohort and protocols that might be developed with family input to ensure easy and reasonable participation? Because it is a single site right now, and we've heard your reasons for such, whether it would be multi-sites or how people at different hospitals could be trained, but also how it is that the family experience could really be taken into consideration to make it possible so that the people whom you need in your studies are the very people whom we can assist to get there.
Okay, thanks for the question, Louise. So in our study, we are recruiting these two cohorts, and the chronic cohort is being recruited first. And I can talk more about that. We do expect, this subacute in some ways will be more challenging. As I showed on the slide, the way that we're defining that is it's pending protocol amendment, but it's 20 to 90 days after injury. Can we recruit this with our current setup as a single trial? We think we can, but we are trying to think out of the box a bit. And in a chronic cohort, I didn't say this, but probably half of the subjects participating in the chronic cohort have come from various places far and wide outside of Chicago. We expect there to be that kind of interest to participate for subacute cohort. The challenge is different though, because it's more difficult for somebody closer to their injury to be able to travel to Chicago. And the way that we have this study set up, the subjects do have to be in Chicago to participate because of we're doing electrophysiology frequently throughout the study, the 16 weeks. So I think that is probably the main challenge that we have for the subacute cohort. And we are providing, assistance with housing and transportation and so forth. But the challenge will be to be able to get potential participants to the site. And while this is a single center study, we're thinking much more broadly about how we're going to recruit. And we have talked to a number of centers about helping us to be, potential referral sources for this study. But, I think the potential is even bigger than that. Anybody who's interested, we'll be doing advertising and so forth. But the big challenge, it boils down to being able to travel to the site. If somebody's living outside of Chicago, and you know, the family and is important in making a decision to participate in the trial, especially a trial like this one, I think. And you know, we're aware of that, but I don't know if that answers your question, Louise. But we are aware of the fact that enrollment of the subacute cohort is presenting different challenges than the chronic cohort for obvious reasons. And we're about to begin enrollment of the chronic cohort. We're anticipating these problems, but we're going to have to be flexible in how we adapt to them, because we do want this to finish in a timely fashion.
Thank you. Right here.
Hi, I'm Maya Hatch. I am from University of California, Irvine and the Reeve Irvine Research Center. My question's pitching off of some of the earlier questions about the outcomes. I was directing this more towards Dan. So in the current one, you said your phase 2 proof of concept, it's one single site. I like the idea of, collecting the motor end plate as well as the other outcomes. Can you speak to the frequency that you're going to be collecting that data, both before the intervention and after the intervention? And that's really related to, typical phase 2, phase 3 trials.
Yep.
Only have 1 or 2 post-assessments that really rely on those clinical outcomes. And then related to that question, when you move into phase 3, have you thought about doing more frequent assessments to capture the variability, particularly with the motor performance tests?
Okay, so there's a lot to unpack there, and I didn't go through the details of the trial, but I can summarize it. It is a 16-week trial, 12 weeks of treatment, placebo-controlled, randomized 1-to-1 as a daily subcutaneous injection. And the reason for the 4 weeks with no intervention, where we're still collecting information on assessments, is because the preclinical data suggests that there could be continued improvement after stopping treatment. I think that's true for a lot of potentially, neuroreparative regenerative therapies, especially with training. If you're making new connections, you may continue to build on that even after you stop treatment. Now, as far as the assessment frequency, for the chronic cohort, the visit frequency there's a baseline assessments. There's assessments at weeks 2, 4, 8, 12, and 16. And for the subacute cohort, it's going to be the same, except we're eliminating the week 2 visit. We're trying to make it less burdensome for the subacute cohort. But as far as which assessments being done at which visit, it really depends. For the assessments that don't take too much time, like 10-meter walk test, 9-hole peg test, we're doing those at all of those visits that I mentioned. For the electrophysiology, we're doing it at all of the visits that I mentioned, and it's a lot of testing. But for some assessments, we're doing less frequent. Like for the motor score, in the INSCI exam, we're just doing a baseline in week 12. And some of the other assessments we're not doing as frequently. But as we move to phase 3, planning for success, it would be a multi-center trial, and the focus would be on clinical efficacy. I don't know that I would do more frequent examinations. I think probably every month, every 4 weeks or so is probably sufficient. But I would think about maybe adding something else to make it more informative, like, and I put this on the slide, a wearable device. This is a good marker of mobility. And there's different kinds of devices that measure different things, but it's not going to be a primary endpoint, but it's going to give you additional information to support. If you show an effect on the most important clinical measures, it'll give you information that supports that the treatment is doing what it's supposed to be doing. And it's the whole time the treatment is being administered and after, right? So it's more frequent. It's essentially a lot of data points.
Thank you. One last question, Christian.
grateful for the fantastic talks, but my next question is for you, Matthew, actually. Dr. Jessica Kwok, who couldn't make it here, was supposed to present something about an oral treatment. Can you briefly summarize what treatment that was?
Yeah, and you can look in the program. It's already approved oral pharmacologic for liver bile dysfunction, I think, in kids in Europe. And apparently her hypothesis is that it's effective for addressing the glial scar. And so there's a company that's formed with backing from a family with SEI in Europe, I think in the UK, and in the process right now of designing a clinical trial, phase 1 proof of concept trial. Yes. There are lots more questions, and my encouragement to everyone is that because I'll want to ask them in the break-in session this afternoon. So please just keep thinking about them, and you can bring them to that conversation later. Thank you, gentlemen. Thank you, Daniel. Thank you, Mikol. Okay, now I want to introduce you to my friend, my colleague, Quinn. Quinn used to be our board member, came to us, I think through the podcast, right? That's how you found us. Oh, so I'll shut up. Quinn, please welcome Quinn. I'll let her introduce herself. Oh, we need microphones. Come on.
Video. All right, perfect.
Yeah, cool. Hi. So I'm Quinn Brett. I'll tell you a little about me, and then we'll talk to Mason a little bit, and then we should have Sam. Yay. Sam will be joining us also.
Thank you, gentlemen. Oops.
So I was a professional rock climber and worked search and rescue in Rocky Mountain National Park. So technical search and rescue from somebody who was hiking and rolled an ankle to somebody stuck on the side of a cliff. My team came and got your ass back to the safe ground and hopefully in safe order. And in 2017, I was injured rock climbing in Yosemite National Park. I took a 120-foot fall, fractured my T12 vertebrae, and landed me here as a paraplegic in a wheelchair. In the hospital time, I was, of course, desperate and going through all of the emotions that do happen with this injury. And I found myself pretty disappointed, frustrated, and just scouring for any information, education about what was happening. Of course, I was desperate for what could return me to my return of function the way I was before. But I couldn't find a lot of that information, or a lot of it wasn't very clear. Should I go spend $100,000 in Bolivia on stem cells? I just didn't know, and it didn't feel right. So as I was scouring the internet, I found Unite to Fight Paralysis. The first thing I found was the CureCast podcast. And I guess what I want to walk you through is, yes, my journey with Unite to Fight Paralysis, but what do we do? Why you're here, yes, we put on the symposium, but we do a lot of other things as well. So we have the CureCast podcast, which I found, and it was in the early infancy stages in 2017. And the first couple episodes I just clung onto, like episodes 1 through 5, walk you through at like a grade 5 reading level, what is happening internally, pathophysiology with the injury. It was fantastic. Having an EMT background, that's the information I needed. Like, what's happening? Why am I bruising? Why is it not healing the exact same way that the peripheral nervous system heals? Within that, I found that we have a Cure Advocacy Network. So this network is our legislative arm of U2FP. So we have passed 4 bills in 4 different states, equaling up to $40 million of spinal cord research money. So this money is allocated per the states. We're trying to pass our 5th in Wisconsin. So if you live in Wisconsin, have friends in Wisconsin, let them know that it's happening. We've been doing this for like, how many years, Mackenzie? 6 years. Let's pass this bill. And this stuff, this bill, this money, like the University of Minnesota, the E-stand trial, all these electrical stimulation trials are funded because of this legislative money. And the importance of that is manyfold, in that the research is not just the nuanced research. We want the repetitive stuff. We want you to find the good thing, and then let's continue on with it and continue your funding so you can follow through with the research. Let's put people with the injury on the table to help decide where the money goes with the research. That kind of stuff. And the most difficult part of this process for Unite to Fight Paralysis is, of course, it's legislative work, it's lobbying. It's not funded ever. So we have our little QR codes on our table, and if this stuff strikes a chord with you, please consider being a monthly donor and or a one-time donor. That would really help any and all of these initiatives that we do. We also have our Lab Rats program. Matthew talked about that a little bit yesterday. So we put these types of interactions that people with the injury have with researchers here at the symposium, creates relationships. And then people with the injury go into labs and help consult, and reminding them of the nuances from wanting to grasp in this way instead of just maybe doing this. Thanks, Jason, for the one laugh. Sam, did you see me? I was holding my finger up. Thanks.
That was great.
And then, of course, we have our symposium. So this is our 19th annual symposium. And this was the genesis for Unite to Fight Paralysis in the first place. Bringing people, stakeholders, from clinicians to people with the injury to researchers, all in the same room to talk, to collaborate, to have cocktail conversations and dream up crazy ideas and then follow through with those ideas. So thank you all for being here, and for many of you for being here many years in a row. Let's see. Speaking of voices, so our voices here, all of our voices matter. And speaking of voices, I wanted 2 voices to be present, or more voices to be present in this. So my work as a development director is, yes, fundraising, but I've noticed there's kind of a gap in our community from people who have the injury, but are so consumed by the secondary issues that it's really hard to do any sort of advocacy or education about themselves and this injury that I was seeking out, to folks who we're told inherently in the hospital that we are definitely going to have to adapt and that we're never going to walk again and we should move on and live life. And so there's this dichotomy between having no hope or false hope, and Unite to Fight Paralysis here, we want to give informed hope. And with that informed hope, we can hopefully get more advocates in our corner. And so my work has been trying to bridge the gap of getting the younger community members, or the people that are still so young and sporty or paraplegic that they can go out and they can live their lives and they can be sporty and do all of these things. But how can they help us remember that we have friends out there that can't go play outside, and that we have friends out there that want a return of function from hand grasp to bladder and bowel, even if that's not your concern. So my ask is to ask Sam and Mason to tell us a little bit about themselves and how they landed here, but also maybe spitball with you all of what can we be doing to bring in more people of our own community to give a shit about this stuff. Go ahead, Sam. Oh, sorry, I looked at Mason, but go ahead, Mason.
Thank you for that. So obviously, as Quinn mentioned, it's not all about walking. And I think the coolest parts about all of this is that we are talking about breathing. We're talking about hand function. What does increased breathing capacity look like in singing, right? Like, these are the real basic things that people with spinal cord injuries want to get back and see it. And so that's super exciting. So thank you for mentioning that. But I'll introduce myself. My name is Mason Branstrator. I am 21 years old. I was paralyzed when I was 17 in a downhill skiing accident, went off a jump, landed on my back, and I'm now a T12 incomplete paraplegic. So since then, I started content creation and became a voice in the community and have reached millions of people. I now have 1 million followers across everything, and I'm trying to find out what my position is in this role. And I'm super honored to be here and be learning more about spinal cord injury research and how I can bring it to social media. We talked about, making a wave of impact on social media, like the ice bucket challenge from ALS. Like, what could we do? What kind of challenge could we do for spinal cord injury research? We talked about the disparities between dementia funding, ALS funding, and spinal cord injury funding. Like, what's that about? These are the things that we need to talk about to really get this thing rolling, you know? That's the noise that we need to create in our community. Sorry, could you guys not hear me too well before? We're here now. But yeah, these are the big things that I'm coming away with. And as a voice in our community, I'm super excited to supercharge this movement of spinal cord injury research and getting people to give a shit, right? That's what it's all about. We heard a lot yesterday. One of the main things that's missing is people with spinal cord injuries being involved in the research. So we know the right direction that it's going in, but also just advocacy work that needs to take place to get this funding to happen. So start thinking about what the next ice bucket challenge is. This is really important, you guys. I'm serious. But yeah.
Sam, thanks, Mason. Sam, tell us about yourself and tell us why you're here.
Sure. Hi, everyone. I wish I could be there in person, and I've met a few of you I know, and still hope to connect with a few of you in the future. So my name is Sam. I sustained my spinal cord injury in 2018 in a bike versus car accident. And I was in the middle of my PhD when I sustained my injury. So I went to Craig Hospital. I met Quinn. Quinn and I go on lots of bike rides together and other adventures. And then the community is small, so I know of Mason and still looking forward to meeting him in person. So I went back to grad school, finished my graduate work, and then really was motivated to make an impact in the spinal cord injury field. Unfortunately, there just were not opportunities in Colorado after I graduated. So I now currently work at a CRO in Boulder for a small biotech company. And I have to give them a little bit of a plug because they funded my registration. They were going to fund my hotel, and then also to high fives for funding my flight, and then also Quinn for inviting me to be here. But just I have to give a plug for the company because this is all about setting up collaborations, and I have really wanted to make an impact in spinal cord injury. So if there's ever kind of a research need, please look into Mosaic Biosciences. But we do everything from antibody discovery, phage discovery, campaigns, basic science, like trying to come up with ideas, all the way down to through the preclinical stage. So creating the protein of interest, cell-based assays, binding functional studies, PK/PD analysis. So yeah, we'd be really excited to get projects related to that. But yeah, I guess that's like a lot about me. I'll let the conversation continue.
Yeah, I guess I wanted to ask both of you, how do you think, I think I asked you this in email preemptive, how do you think, what would be useful for more of our community to grab a hold of Unite to Fight Paralysis, or just being curious about the research landscape, Sam, or just educating themselves on the injury in general? Do you have any ideas?
I can go ahead. Educating yourself on the injury in general. I mean, I think that so many people look at the ways that we're forced to adapt, and that's what we move with. But a lot of the stuff here, we're talking about how can we get some function back. You know, when David mentioned to Gabe yesterday about the breathing, what might that do for your music? Like, how much longer might you be able to go? That was something that he might not have thought about before. And so we're looking at new innovations to really take our functioning to the next level in some regards. But I think in terms of my own function, there's a lot of stuff that I would love to see some more research on, you know, sexual function, bowel and bladder stuff, bladder spasticity. those are my biggest issues. Those are the things that make it hardest for me to show up as an energized 21-year-old that's trying to make a difference in this world. Like, that's it. It's not the bigger stuff, you know, it's just like bladder spasticity and some weird internal stuff that I think might be related to my spinal cord injury. Like, that's what I would love to get looked at. And, you know, I think there's a lot of those slight things. Yesterday, when we saw the blood pressure regulator that took that woman from being homebound to having this wonderful life, those are the types of innovations that can be life-changing. So, yeah.
Anything to share, Sam?
I think that I've seen a lot more involvement between like having lived experience, folks connected with scientists and programs. And I think that's really a great way of giving people a voice and also making sure that there's consideration for people's priorities, because they are different than walking, as we've heard a bunch of times. And a lot of people don't know if that's conveyed. So I think those programs are really helpful.
Does anyone have any questions for Sam or Mason out in the audience? And if not, no worries. Yeah.
Hey, hang on. Mike's coming to you.
I can repeat it. Say it, I can repeat it.
Hey guys, my name is Matt from North Idaho. I'm also a young person. I had my injury 2 years ago. I jumped into a foam pit and didn't get out. Well, I got out, but you know, not with legs. And ever since then, I started to post on social media as well. And I saw a big gap between like the younger generation, even like, you know, older people thinking that this is just how life is going to be and we just have to accept it. And I've always been super against the acceptance word. I think my parents taught me to be contrarian and like, hey, you're in poverty. Are you going to accept it? No. Hey, you have mental disorders. Are you going to accept it? No. So I think the big gap is the acceptance word. I think there's a lot that us young people can do. And I'm just wondering from both Sam and Mason, what you guys think us as young people can do to rally each other and even to get people kind of, you know, for lack of a better term, off their butt to start talking about these things. And I think, you know, advocate for more money, for faster research, and even just for us to, you know, rally together to inspire the next generation. So what do you guys think we can do?
I just want to double down on this ALS Ice Bucket Challenge. I'm so serious, you guys. We need a global wave of a trend. We need people to get excited about spinal cord injury research. We need them to see what our lives are like and how they can be better and where that money could go to. So anyone, like, please think on this. We're serious. It needs to be something everybody can do. And if we get this train moving, I don't think it's ever going to stop. So that's my 2 cents.
I want to ask a more pointed question to you, to all of you, really. I'm not sure all of you in the room understand some of the history, and I also would confess that I might have a bias. Quinn and Jason, you two represent kind of a breakthrough in our organization of recognizing a history of that divide between people who are focused on care and those who are focused on cure. And more importantly, a divide between people who, like, I'll give you an example. Years ago, a former board member of U2FP said, who plays quad rugby on the team in Pittsburgh, I believe, was like, I can't get any of my guys on the team to even care about what I'm doing with U2FP and even care about research. They're just too busy, like, well, I don't have time to think about that crap. And I think of you two, Sam and Mason, active people. You know, you said you and Quinn go out on rides. Quinn, you go out on rides. You and Jason did the rim to rim. Quinn, you did the divide. And you even had flack on social media for like, oh, like, so you can go do your active stuff. So and I can't. And so that's why you don't really care about cure. Right? So there's this weird history. And so I want to ask all three of you, just in a way, if you could comment or address that, like, how do you live that? How do you experience that? I mean, you're not high-level quads, right? Which is a big distinction in our community, right? In terms of what you're capable of doing. But also just this, like, I can go out and ski, or I can go out on a trip, and I can go ride, and I can do this and have these adventures. And yet you still care about the science, and you still care about curing paralysis, and you still care about an ice bucket challenge, Mason. What's different about you and some of your colleagues and friends and element of that community?
I'm just going to say right off the bat, we are privileged to have that amount of energy to put into this research and being energized about it and being a part of it. And I think there's, like, the more problems you have to deal with, the less time and energy you have. And all of those people that are like, man, I got too much of my own stuff going on. You're trying to talk to me about, getting in here and thinking about a cure that we don't even have concrete evidence that what's going to happen. In comparison to infrastructure, you talk about accessibility. Can we make that building accessible? Yes, we can. Here's exactly how we're going to do it. Can we get you to walk again? We're not sure. And so it's more concrete. And I also think we are privileged to have the time and energy to be here. And thank you all, everybody, for being here. I'm just blown away by the amount of support that spinal cord injury research has moving into the future with all of you able-bodied people and all of us coming together. I mean, it's just incredible. So thank you all for caring so much. Yeah.
Jared.
Hey, all right. Hello, everybody. My name is Jared Potter. I'm from Salt Lake City, Utah. I am just under 1 year from my injury on October 5th, where I decided my life was too boring and I ran into the side of a hill with a paraglider. But anyway, here we are. I feel incredibly privileged to be able to be here with all of you and to learn so much from the community here. It's been, I think, a difficult coping process to go from, what my life was like before versus what it is now. And, I'm not sure that I really like it. But what I've found to be the most useful thing, honestly, is being able to meet other people in chairs and to learn from their experience and how kind of they've coped through this experience. So I'm kind of curious to know from all of you, all three of you guys, what was probably the main insight that you gained during your recovery? And that doesn't have to necessarily be within the first year, but just any time over, from your injury till now, of what was the most important insight that's helped you cope with this experience? We'll make Quinn go first.
Make me go first? Oh, I wanted to hear from Sam. She might have to think about it. Yes. For me, golly, the biggest insight. For me with this injury, I guess even before my injury, I practiced a lot of yoga and would do a lot of crazy adventures and runs. For instance, I ran the rim to rim of the Grand Canyon and my knee was in pain 87% of the time. But in my head, I just kept saying, this is how it is now. In 5 seconds, it might hurt, but it might not. This is just how it is right now. And so I think that's my attitude with spinal cord injury is the same. This is how it is right now. And I'm hoping that more of our community can see it that way. This is how it is right now. It's not hopeless. There's things that can change. There's things that we can get back. And so, yeah, how do we shift our community to think it's not hopeless? This is just how it is right now.
Sam.
I could use another couple seconds if you want to go, Mason.
Okay. I just have to say, we talk about care versus cure. It's always a both and, right? We needed a tremendous amount of care to show up as our paralyzed selves today. But how do we take that and use that energy and move it into the future and ask for more and see what that looks like? So, but I think the biggest thing that helped me cope was that I had to accept exactly where I was, like Quinn said. And I think when you accept exactly your situation so wholeheartedly, you forget that things can change too, because you're forced into accepting this new normal. And this is just how it is now. And so sometimes you lose hope in the future when you accept things.
Yeah, but we also have to keep that like fire of dissatisfaction. So
Yes.
that's like why we're here. Those of us with the injury, and maybe those of you who are researchers, like there's still a dissatisfaction of not solving this problem. Let's go.
I think for me, really finding my community is what gave me the most hope after my injury. And that ranges from adaptive sports to then also the scientific area. And I've just found that like, I'm someone who very much trusts statistics. So when I was told I wouldn't walk again, I was very accepting of that. But there's a lot of things that I found that my friends are doing that's I never thought I could do. And then I try it, and that would really give me a lot more hope. There's just a lot of people pushing boundaries, and it's exciting to see, and it gives me hope as well and forces me to do it too.
I totally have the microphone, so I think I'll ask a question. So, and it's, Sam, this is Candace Floyd. I don't know if you remember me, but we worked
Hi.
together. We worked together. And so it's a little bit of a confession, and it's also a question. And so we had a background, I don't remember how long ago it was, maybe 5 or 6 years ago. We worked together, the three of us, on some proposals, and it was my first attempt to include persons with lived experience in the research process. And my confession is that I think I did a horrible job at it. And so the three of us met, and basically how I did it at that time is I presented ideas to you guys and got feedback, right? And we've changed that approach completely now in having Plex-driven parts of the research program. So I think I've learned a lot. I think our teams have learned a lot. But I just wanted to hear some things maybe that from both of your experiences, Quinn, and about what we did wrong then, what your perception was, and what people can do better now to have a meaningful avoiding tokenism connection between Plex and research.
That's a good question. I don't know, Sam. I think I felt Sam was actually more of the expert because she's in this field, and I was still also new spinal cord injury at that time. I think it was only 2 or 3 years in. But it did feel not as participatory or communicative, I guess. Like it was like, hey, we need you for this. What do you think? But we don't know what we don't know. And so I guess I would have wished for a little bit more roundedness of like, having you not done it before, but now having that experience, can you convey that on the front end? Like, here's how we've done this before. And if you have questions, we're always obviously open to those questions. And here's other ways, I don't know, I guess has to be more interactive and problem solving together. How did you feel, Sam?
Yeah, I'd say it's kind of tricky, and like everyone's involvement might look different. Just because like for me, I was really wanting to kind of like dive into the science more and follow through with that in terms of hearing about it. I don't know that that's everyone's goal. Yeah, I guess it's a tricky question, but maybe more like consistent communication and just hearing how things are going. But also to Mason's point, like we also kind of also have limited time and energy. So I don't know a good answer at this point.
One more question.
I'm just going to say real quick, I want you guys to know that I'm not the brains behind the ALS X Bucket Challenge and transitioning that to spinal cord injury. Jason is. He'll be coming up here later. So yeah, he's the brains behind like this operation right here, just so you know.
Hi, my name is Mike Uppenkamp from San Diego, and I have a son with a C3 AJA complete. So listening to this, I'm feeling that everybody in this room has different roles. So I'm an SCI dad. I care about my son. So I'm here looking at the research of what can help him walk maybe in his lifetime. But he's at home surfing, mountain biking, going to college. He's got this community that follows him. And I think it's the promotion of what people with injury can do that will give us more attention in mainstream world. So that's kind of where I'm at, is helping people get out there and get injured again if they want to. You know, go surfing, paragliding, off-roading. We do all kinds of stuff. And that gets the community's attention. And if we have the community's attention with 1 million followers like you have, then we have a voice, and that voice can get out to many more people. So that's my thought on it.
Happy?
Yeah, I just want to, can I wrap it up?
Sure, I was going to have one more ask of just remembering, we do have, I know I'm the development director, so I'm the fundraiser gal. You're going to have to listen to this. But this month is September, Spinal Cord Injury Awareness Month, and we have a private donor that has offered up $100,000 match. So.
And we're at about 50.
Yeah. So just
So-
consider, this is a huge opportunity. It would like double a lot of our programs. Thanks.
Well, then let me use that as a wrap up 2 ways. One, I want to execute a challenge kind of to everybody in the room, given what we've just talked about. And it's a metaphor that I really love when I discovered it. Do you all know what the overview effect is? Do you know that you've ever heard that term? Astronauts there's, I don't know, 14 or 16 astronauts who've gone to the moon since the 60s. And they all have had a very similar existential, emotional experience. They look back at this planet, and this is called the overview effect. They're like, oh my God, there's no borders, there's no race, there's this blue ball in the middle of black space, and they all have the same feeling. All the shit that's going on, we can do better. And I like that metaphor because that's, I know personally my experience, and many people with spinal cord injury, it's like being thrown out to space. In this instant, you're thrown out of your orbit, you're thrown out of oxygen, you require a high level of technology to survive, and every part of your body is affected by zero gravity. And you look back and you see things differently. And so my challenge to my community is that you have a perspective that the rest of the world does not have, and they need to hear it from you. You need to speak up. You need to be part of the activity that we're talking about. And to everybody else in the room, I would challenge you, scientists, clinicians, companies, you need our perspective. You need that unique perspective, and especially in one area when it comes to healthcare in this country. We have lived through the underbelly of healthcare in the United States. I mean, I see heads nodding. We know what it's like really in the real world. And there's often a disconnect between what scientists are doing abstractly in their lab and how it translates to reality in the wild west of healthcare in this country, or the world for that matter. That's a very important thing, I think. So I lay that challenge to you all. It's a reason for why we have much to offer you. And our community can be empowered by that. So we have to, that urgency that you asked about, Barry, yesterday, that urgency has to come from us. The second thing I'll say is back to what Mason said, it's another plug for money. All this stuff is highly nuanced. If we wanted to raise money to get a curb cut out in the intersection near my house, it's easy, it's black and white. Let's do it. On February 15th, it's done, it's paid. When we try to raise money, it's like, well, it's really kind of nuanced. It's really complicated because we're trying to affect the research ecosystem and we're trying to bring people, and we don't get funded. We don't get our grants funded because we're not like buying 5 monoskis that are going to be
Which
used
put
on
12 smiles on
Yeah,
people's
right.
faces.
Yeah,
We can't
exactly.
It's not as
We're
tangible.
not doing these like material things. We're doing highly nuanced, like strategic advocacy stuff, and it's really difficult for us to get money. So this is a little plug for why it's important that you consider donating and supporting us. Okay.
Thanks, Sam.
Thank you. Thank you, Sam. Thank you, Mason. Thank you, Quinn.
Thank you, everyone. You guys are awesome. Thank you all for being here.
Oh, couple announcements. So remember, we have a survey. I'd like you to fill it out. We're going to do posters, correct?
No, we're doing my voice.
Oh, are we?
We're doing my voice. right?
Oh, that's right. Yeah.
Yeah, I thought you were segueing.
That's a good segue. Perfect. Well, then let me just one more thing. For those of you who are going to still be around, I see some suitcases out here. I'm sure you got your flights to get to. Whose idea was it? The flying V? Is it your idea? The flying V? The V, the wheelchair V. All right, so I just want to let you know real quickly on the next break, everyone who's willing, who has a spinal cord injury, would like you to go out to the lobby. Matt is going to film, or Ryan's going to film all of you with that cool social media Gen Z tool oscillating. And we're going to do a flying V coming out of the lobby away from the escalator. And then the rest of you, if you're willing, we'd like to have you all line up, and then Ryan's just going to go all the way down the line. And we want to use that as a thing that Matt's going to put together to promote the symposium next year. Okay.
We're to the My Voice section
Yes.
of this program.
So now we'd like to hear from everybody.
So we'd like to hear from everybody. And it was great segue. So you guys are great. So I think the format here is I need help. So Emily, I need you to go get the xylophone, please. Bring that fancy $30 instrument into the room. And then we need a timekeeper. It might be me. And the idea here, as we said, and Laura and I kind of, I'll give Laura credit for this. This is Laura's idea transformed through the lens of 50 people, but is to take 3 minutes and give that perspective that you want everybody to hear. Would you like to give one? Am I putting you on the spot? No, no, I think we can do it from right here if you're okay, unless you would like to go up, and you can. So it's up to you. Okay. Would you like me to hold the microphone, or you got it?
Okay, let's try.
Okay.
Let's see if I can keep this on under 3 minutes. My name is Laura. I am a longtime live streamer, first time attendee. I'm coming to you from San Diego, California, where I worked up until the time of injury for 22 years in the biotech industry as a scientist. And I'm also a lifelong competitive athlete. And so when my injury occurred, those were two things that I felt like I can't really do anymore. So my best thing to do with myself is to make an experiment of myself. So where do I even want to start? So the best way I found to do that was kind of to throw myself into activity-based therapy. And being in San Diego, I'm quite lucky to have quite a large selection of activity-based therapy centers, one of which is here today, Renew Health in the room. And also at the time, I had the opportunity to participate in several clinical trials, one drug study, one transcutaneous study. And while there were a lot of really good things about that, immediately I was had ideas of how they could be run better. And at the time, the Lab Rats initiative had just started happening. And of course, that was an obvious, of course, I'm going to do this. And that is how I met Matthew. And also I attended the live stream when Candace was speaking in Salt Lake City. And I was very impressed with her ideas. So I joined Lab Rats. And through that experience, long story short, I found out about Dr. Uzma Samadani doing the epidural stim implants off-label. And again, I was like, well, yeah, that's absolutely what I'm going to try. 2 years ago this month, I went to Minneapolis, had the stimulator implanted offline. I went back home to San Diego with my little device. This is not part of a clinical trial. Mika tossed me on the table. We turned it on. I immediately kicked her right in the face because we had no idea what we were doing with it. It really truly is the wild, wild west out in San Diego. Been working with it for 2 years, trying to figure out the best way to get the most return possible using ABT plus the stimulator. We've done awesome stuff, but we both agree that it is not enough. And I'm, of course, awfully interested in the Epi-Stim combo trial that Candace was running, that's supposed to be running. So I said, Matthew, can you get me a place in that pig lab? I'm really interested in being a part of that. And that happened. And I had a phone call with Candace, and it was just a barrage of all the stuff she had going on and all the things she needed and all these great ideas. And could you help with this, that, and the other? And I said, absolutely, I'd be happy to. And a couple of months went by and a couple of meetings got canceled because Candace was just so busy that she didn't have time to meet with me. And finally, I polished off my biotech resume and I just sent it to her out of the blue. And I said, give me a job, hire me, and I will come, I'll help you. And she did. And I've been working in her lab, both as a Plex and as an employee for the past year, doing grant writing, data curation, anything she needs remotely from San Diego. So I came out here this weekend. I got to meet the pigs. I got to meet her staff. I got to see the whole lab space. And it's really been an awfully incredible experience. And so I am just, the way that she has included me and asked for my voice on this combo trial, not only as a Plex, but as a person living with the stimulator for the last 2 years. She's incorporated not only my voice, but the advice of my activity-based therapist, because that was my first question for her. What kind of activity-based therapist is going to be available for these pigs when you implant the stimulators? Because that's going to be the most important part to get that to work. And she's just been incredible to work for. And all we are waiting for now is some billion-dollar company, medical device company, to find us a handful of stimulators, and this project is ready to go. And I just want to thank her for the opportunity that I've been given.
Who's next?
Don't be shy. Oh, of course, Jason.
Jason, I'm coming. Work on my cheating. She won't have to run, I can go.
Hi, can you hear me? Okay. You guys hear from me a lot, so I won't talk very long. And most of you know my background and story and all that stuff. So, but the thing I wanted to say, since it seems to be like open mic time, is I had a realization during this symposium that I haven't had in other symposiums. And it's an appreciation for those of you who are here that are researchers. Can you, if you're a researcher doing work in spinal cord injury research, can you raise your hand or stand up or do whatever? I just want to see like the percentage of folks that are actually doing the work here. Yeah, I mean, yeah, give them a hand. What I wanted to say really quickly is I really appreciate that you guys are here. And I feel like this would be, if I was a researcher, I feel like this would be a very intimidating conference to come to. Because I feel like we're trying to like get our community to speak up and be critical and be a voice and say what we need, right? But sometimes when we do that and when we embrace it, I feel like sometimes it can come off as like overly critical and maybe even berating of what you guys are doing. Like, change this, you're not going fast enough. That's the wrong priority. I mean, I would think if I was a researcher and I came to this conference, I'd be like, I feel beat down. And at the same time, I think because you guys are here, especially those of you here that like weren't even presenting, you're just here to be with us and to learn things. Thank you so, so much. And please keep coming. And yeah, thanks, Christian. I mean, it's true. It's a feeling I got this year that I really felt like I wanted to express to you guys. And I know that those of you who are here, it's because you do care about our voice. You do care about this area that you work in. You do want to see this functional recovery in our population. And I just wanted to say thank you. I hope that's under 3 minutes, but.
Yes, it is under 3 minutes. And I just want to add, thank you, Jason. I'm not going to name any names, There are a few scientists that I have invited many, many times to come to this meeting, and they have said no. Because I don't want to be in a room full of a lot of people with spinal cord injury, and I don't want them, like a couple of them have said, I don't want their moms running up to me with an MRI and like, can you fix my kid's spine? You know, like, I'm like, that's not what's going to happen. But they still will not come, even last year. So, Lana, is there something you wanted to say? Are you sure? Well, can I say it? Lana, I will, but Lana just texted me and said, it is an intimidating conference. Lana is a scientist at Gladstone, and she said it is an intimidating conference as a scientist. Sorry.
Hi, guys. My name is Jessica. I'm actually the caregiver and wife to a quadriplegic. And this conference has been, I also have a science background and in radiation. So this has been quite eye-opening. I liked listening to a lot of the scientific stuff. But really part of the reason why I'm standing up is I know some of you are here, not part of the actual research part, but you're actually a spinal cord injury. The other partner to a spinal cord injury would be your caretaker or your husband or wife. They are somebody who, don't get me wrong, takes some of the biggest brunt of your frustrations and some of the things that go on. And to be honest, some of it is pretty shitty, literally. But on top of that, I'm just part of a group called WAGs for SCI. So we are a huge group of wives and girlfriends of spinal cord injury people. I don't like to call them patients. And we're a big community of podcasts. We do fundraisers to do date nights. And it's also someplace that I learned and I use every day that you can talk to somebody about, which is huge. We now have a group where we can talk to somebody who's had those experiences and something that they can do. And I just wanted everybody to just kind of be aware that even as a caretaker or a husband or wife, there is some place for you to go to get some of that help. We also initiate trying to do some clinical trials. We hear from each other about what our husbands or boyfriends are doing and try to get them involved. So it's also another place where we can share ideas too. So I just want everybody to be aware. I do have a couple flyers with me if anybody wanted to see them or anything like that, but just kind of a little plug just for the caretaker.
Grabbed it from the table. It's hot, it's hot.
Oh, okay. So my name is Wendy Gilchrist. I have a son that he says he's C2, C3. The doctors and us will say C5, C6. But so since the beginning of the spinal cord injury of my son, my husband said, do not cry in front of him. You cannot cry in front of him. So maybe it happened like 3 months later. I was crying. I went to the bathroom and my kid said, what happened? I said, oh, no, I'm not crying. And they said, Mom, if you're not crying, we are going to think that something is not okay with you because you really care about me. So I have another son that he has schizophrenia. And we have had a group of 25 families meeting every month, just like a very unique group, people that just have experienced schizophrenia, we are them for there. So it's very different because with schizophrenia, nobody talks, you are not proud of your people, with your son in the wheelchair that he's doing a little bit more and a little bit more. But at the same time, as you were saying, moms and wives and also friends or girlfriends, they have a space, but not dads. And in this group of schizophrenia, we have dads and moms, and it's really, and friends and caregivers. So I invite all the fathers and moms and friends to come to the group. This is like, we're not doing anything extra, like a big thing, but it's just holding a space once a month for families of people that we care. Because yes, it's a lot of work, but it's blessing. I have met so many angels through out this. I am so blessed to be here. And thank you, everybody, to be part of this community.
Thank you.
My first name is Christian. I'm a critical care physician. And through the experience of being a dad of a 27-year-old with C4 Asia A, I have joined and been blessed with meeting Matthew and Jason and Quinn and Sam and be part of this fantastic community. I want to dispel anybody's fear of being inadequate in this community by pointing out to me as a lightning rod of how constantly inadequate I feel vis-a-vis the challenge of the spinal cord injury, both as a father and as a physician. I am constantly petrified of my own ignorance. Nobody in this room should feel alone in being intimidated or feeling down. I have been at the bedside of my own son following a traumatic spinal cord injury and being a critical care physician at not knowing the very few standards that I already established in this line of work that were not applied in the acute phase. And I didn't have a voice because I did not know better. And I'm here to hopefully be part of the solution and to hopefully help spread whatever the little we may know right now clinically to become standard of care, to give a voice to the rapid responders out there, Louise, and to give a voice to parents and communities, to help my fellow physicians do better, as I myself need to do better on a daily basis. I left this room and this awesome meeting, day 1 of 2 of this awesome meeting last night, angry and very scared because I realized, even though I've been for almost a year with U2FP, I realized how not within grasp, pun intended, a cure is. And I think ignorance is not knowing that you don't know. Ignorance needs to be dispelled by learning from others how much is that we don't know and what the steps are to remedy that. I think there's something much worse than not grasping a cure right now, and that's the risk of isolation and intimidation. And if there's one cure in what we're experiencing here, is the cure to isolation. So amen to this meeting.
2 more. Mackenzie.
Hi, my name is Mackenzie, and I do know many of you in the room, but just as a little bit of my story, I've been in a chair for roughly 10 years now after a fall in 2014. And initially, after my hospital stay, I did go to a nursing facility, a skilled nursing home, and I lived there for the first 3 years of my spinal cord injury. So it was an incredibly dark and lonely time for me. And a few years later, I kind of stumbled upon U2FP through a friend of mine at one of the hospitals in Wisconsin. And I really want to just express my gratitude for this organization and for this symposium. I told you last year when I came, it was my first time coming, and I felt for the first time in my 10 years of being in a chair that I really found, not only you guys helped me find a voice within my state, and also just a home within my community. So thank you guys for that.
Yeah, Mike Uppenkamp here again from San Diego. As a parent, as a dad. So last night after meeting Christian, I launched a Facebook group called SCI Dads. So go on Facebook, look up SCI Dads, join. Let's see where we can take that. Compete with the WAGs because they gave me some input on how to do it last year, but I've been kind of reluctant to launch it. And since I've been an SCI dad, I've helped a dozen families navigate with blink of an eye and other foundations. So if you hear about a person that is newly injured in San Diego, the West Coast, let us know because we can help them get the care they need and answer the questions and be there to give them a hug because we've been there.
Thanks, Mike. John?
Hello, everybody. My name is John Martinson. I'm from the great state of Wisconsin. And I just want to say I love everybody here who's up for the challenge of finding a functional recovery for everybody. I know it affects family lives and there's a lot of stuff, bad stuff that comes with the territory. Another thing I'd like to say is, Mason, with your saying of get up for the challenge, I think you're stronger than what you think you are. And like Quinn was saying, you got to live in the now. And then as far as I feel that if you stay positive and you be active, you don't have a chance to think of the bad parts of what's going on. And anyways, I'm really proud of being in this community, and hopefully we can get a bill passed in Wisconsin for spinal cord injury research. So thanks everybody for being here, and have a nice day.
Thanks, John. And you all should know, John is why we're in Wisconsin trying to pass the bill. And not only that, John is a relentless, I mean, he's a beast in blocking doors and hallways and getting into legislators' offices all by himself and trying to get them to sign on to the bill. He's just been a monster. And that's how close we got last session, thanks in many ways. I mean, Mackenzie and many of the other advocates, but John is just relentless. So thank you, John.
All right, last one.
All right, I'll make it the last one, I guess. I'm Dan Edwards from Coeur d'Alene, Idaho, up north, close to Canada. I just want to say there's power in prayer. And almost 2 years ago, when my son had the accident, I turned to the Lord and I did much prayer and fasting. That we'd find an organization such as this. That we'd find scientists and researchers, and we pray for them. And I ask all of you to pray. Turn to our Heavenly Father and pray for these scientists and researchers that they'll discover through education and through their mind that the Lord might put something upon their knowledge that they can bring forth these cures and these different things that can help each one of these individuals that have been impacted. And I think it's so amazing to be out here, to be a part of this experience and seeing these awesome individuals. And the first person we saw was Mason when he came through the door at the hotel here. And just the bright and the smile on your face, man, it was just so enthusiastic. And you know, now some of the other guys that we've been able to share some time with here and get to know, you guys all just have a special place in our heart. And just, I tell you, keep your spirits up. That's the best thing you can do. Stay positive and keep moving forward. And there's power in being positive. There's power in healing of being positive. And most importantly, turn to these groups. If you have a strong family, turn to your family and look for help with them. As far as the political side of things, I have a political background. I was the mayor of my town just this last January. Retired out of that, be able to take care of more things at home. And I'm all about pushing forth legislation and helping with the cause. So if any of you guys have any push or anything like that, I'm coming from the great state of Idaho, and we can push for legislation there as well. I just need some direction of what you guys would want to push. So with that, I'm grateful to be here. I'm grateful for this organization. Thank you, Matthew, for putting this organization together. Proud of you.
Thanks, Edwards family. I want to give you one more metaphor, and then we're going to go to the poster session. It was something, I think it was something you said, Christian, and also back to you, Lana, about this discomfort and about the isolation and running away from intimidation or running away from or wheeling away from tension. And so my staff probably roll their eyes right now because I have this little mantra I've been using for a while as a metaphor of effective tension. The only reason why I can stand right now, right here, is because I have muscle groups in my legs that are in opposing tension that allow me to stand up against gravity. There's a reason why we have life on this planet, because the earth revolves around the sun and is held in tension, gravity. The way forward for us collectively is to be held in tension, to be honest, to speak the truth about what we see, and it may not be welcome. You might feel discouraged by what you hear, but the truth is better than false hope, as Quinn said earlier. That informed hope often requires an effective tension. So scientists listen to us and the truth of what we see, we listen to them. We listen to Mikol Lauw, who says, unfortunately, the customers for Lineage Cell to get to the clinic require insurance companies and investors and physicians to do those things. Those things are sometimes difficult to hear, but that effective tension is what holds us up and gets us forward. Without it, if we run away from it because it's uncomfortable or intimidating, then we don't get where we need to go. So just give you that additional, I like metaphors, so I apologize. So I'm stacking them up on y'all. Okay. Posters. We are going to go in alphabetical order. So I hope someone has talked to everybody about who's coming up. Is that you, Jason? You're first?
Yep.
Come on up. Please welcome Jason Biundo. Yep.
Yes. Everyone hear me?
Yes. Sweet.
It's my clicker. So everyone was just talking about their voice. My voice is that I'm a little bit of both a researcher and a person with an injury. This is my second time giving a talk at UTFP. First time I talked about advocacy and getting everyone involved in the research process. Now I'm going to be talking about a little bit of some science work that I've been doing the past few years. I'm a first-year PhD student at Harvard Medical School. I just started a few weeks ago. And today I'm going to talk about some work I've been doing the past few years before I started my PhD. So we're trying to create a new quantification system that is open source that the whole community can use for quantifying locomotion recovery after injury in rodent models. And importantly, we're only looking at hind limb recovery so far. So our initial goal for this was we were trying to screen multiple different treatments to test their effectiveness in treating spinal cord injury, but the current standard for measuring locomotion recovery is usually the BMS score, which I think most of us probably know has its own disadvantages. Pretty much you watch mice walk around in an open field and manually test like how well they're walking by manually observing them. But I think that leaves room for a lot of bias, and some parameters are vague, and some labs do their own scores differently. So we want to create automatic system to do that in an unbiased way. There have been some commercial approaches and some labs have their own fancy setups. But I think some of them have a drawback is, for one, they're super expensive, so not everyone can access them. In other cases, the methods for how they work are hidden, so you can't see exactly how they're working, or they're too complex to actually reproduce. And so I think what we're lacking is an open source analysis platform for doing this kind of quantification. And so we can effectively, unbiasedly monitor locomotion recovery. And so our goal is to develop an improved scoring system so we can compare different types of injuries and also have it be useful for the whole community. And so our setup is very simple. We pretty much watch mice walk back and forth basically an enclosed plastic chamber, and we have a mirror underneath it so we can get the side view of the mouse and the bottom up view at the same time with a single camera. It's very easy to build, and we use an open source program to track the mice. So it's called Deep Lab Cut, and what it does is it uses AI and machine learning to track a lot of different points on the side view and the bottom up view at the same time. And I trained the model to get really good at this, and it gets within a few pixels error. So it's really effective at tracking the mouse's locomotion. And we track them moving side to side. And from that tracking data, then we can develop a pipeline to calculate different parameters to describe how well the mice are moving. And so to do that, the mice have to be at least moving a little bit. So all mice, at least with the thoracic injuries that we use, they can at least move their forepaws, and they slowly develop the ability to use their hindpaws as well. And we calculate the steps by using the velocity of their paws relative to the rest of the body, so we can automatically find when they're stepping. And then around those stepping, we can calculate different parameters describing how well the mice are moving. So we can calculate like the angles between different joints, how high their body parts are off the ground, the rotation of their paws as they're stepping. We get a really good overall picture of how well the mice are moving. In total, we calculate over 75 different parameters, and we can separate them into different categories like hind limb flexibility, coordination, body weight support, stepping, parameters related to their tail, things like that. So we can get a really complete picture. To look at all those parameters at once is a lot. So we could look at those parameters individually and compare them between different treatment groups, or we could try to plot them all at once in something's called a principal component analysis. Essentially, it just combines all the parameters into one point. So each little dot on the screen is one individual trial of a mouse moving from side to side. And the bigger dots are an average of all the trials for a particular group. So in the top left, you see that's what all the mice look like at week 1. And then the bottom, you see that they're starting to move towards the baseline on the orange. So the green and blue are the controls, and the purple and red are some of the treatments that we were trying. And you can see that they're moving closer to the baseline group or towards the intact group as they get their treatment. And the cool thing is we can also separate out all the parameters that we're looking at all at once to see what individually, like what might be most important. And so we find consistently like the height of different parameters is one of the most important things, like the height of their hip, for example. We think that's a good correlate of body weight support, their coordination of their steps. Are they moving their left hind limb followed by a right hind limb, things like that. So we can get a very detailed picture of what exactly is changing. And we can also make some cool visualizations. So we can do the classic stick view diagrams, which is just an overall picture of how the joints are moving. And what we can also do is use the bottom up view that we're getting and get like a footprint analysis. So you can see how well the mice are walking. You can see in the first week, they're dragging their paws a lot. But with the treatment, they can start to take some steps, and you can see like how long they're taking between different steps. And it's a really cool overall visualization. I don't have time to get into super detailed, but overall, I think we can reliably calculate different parameters describing the locomotion across different severities of injury. And we've also tested it for different CNS injury models. So we we use Parkinson's models, an MS model, and overall the system is really cheap and easy to use. I've built a detailed protocol for how to set it up. I'm writing a preprint now, so that will hopefully be live soon. And you can email me or Zhigong that I've been working with to get access to the pipeline. And thank you. Thank you, everyone.
I just want to say congrats on that work. It's really important to have unbiased agnostic approaches to these kinds of things. Another model that we use is something called MoSeq, which is a way of looking at how a mouse moves and uses its natural behavior, things like grooming, exploring vertically, that kind of stuff. And that gets to the idea of using natural behavior as opposed to learned behaviors. So I don't really have a question. I just wanted to say congrats. It's a great piece of work and it's going to push things forward quickly. Well done.
Thank you. Thank you.
Hello, is this on? Oh, yep. Good afternoon. My name is Sydney Carpentier, and I'm a fifth-year PhD student at the University of Minnesota. And today I will be presenting on aim 1 of my dissertation project, which is entitled Development of a Novel Evaluation Scale to Measure Mental Body Representations in Adults with Spinal Cord Injury. So mental body representations, also known as MBR, consist of body awareness, which is the generation of conscious awareness of the state of the body, how different body parts relate to each other, and whole body awareness, as well as visuospatial body maps, which is the conscious awareness of where the whole body is in space and it updates in real time. Adults with SCI have deficits in mental body representations, which is important to know because it can contribute to the prevalence and maintenance of neuropathic pain, as well as impact sensory and motor function. Although we know that there are deficits in mental body representations in adults with spinal cord injury, there is currently no spinal cord injury related specific scale measuring these quantities of mental body representation deficits. So the purpose of my dissertation will be to develop a reliable and valid evaluation scale measuring MBR in adults with SCI. Specifically today, I will be going over the item generation stage of this project. Later on, I will conduct the reliability and validity testing of these new items. The initial item generation was completed by a therapist who specializes in cognitive multisensory rehabilitation, or CMR, which is a therapy that is aimed at increasing MBR in the brain. And this was presented by Dr. Ann Van Winkle yesterday in her talk. Specifically, this was the therapy where we saw the participant with their eyes closed and the therapist asked them whether one foot was higher or not. So after those initial items were generated by the physical therapist, we sent those to 7 Italian PTs and 3 Brazilian PTs, also specialized in CMR, and they reviewed and gave their feedback on those items. Importantly, a next step will be to gain feedback from adults with spinal cord injury with lived experience on both the content of these items as well as the images presented in the items. And I'd also love feedback from physical therapists who have experience with and without CMR training in the United States. So what does the scale currently look like? There are 6 items. Item 1 is body dimension, and the goal of this task specifically, I guess I should mention that there are multiple tasks within each item, but given the time, I will just give one example of each. So the goal of this would be to correctly identify the length or dimension of a given body part. Specifically, the patient is asked to identify how many of their own hands will it take to get from the pelvis to the top of the knee to get the length of the upper leg. In item 2, it focuses on body awareness and awareness of spatial relations. Specifically, this participant is asked to identify with their eyes closed when the participant moves their foot, is one foot closer to an object or their feet even, or is one other foot closer to the object. Item 3, spatial awareness, asks the participant to correctly evaluate where the body is in space. This example, the physical therapist has their arm in a certain angle and asks the participant to look at their hand, and if their hand is to move in that exact same direction, would it miss their knee or touch their knee? Item 4 is sound localization in relation to a position in the body. So for this, the physical therapist will snap their fingers to make a sound in specific locations around the participant's body. Specifically on the left-hand side, this physical therapist is asking, is the sound coming from the front or the back of the body? And on the right-hand side, they're asking if the sound is coming from the left or the right-hand side of the body. They'll also be asking at what level or position of the body is the snap coming from. Items 5 and 6 are both self-reported items that may look similar to other tasks given to adults with spinal cord injury, to color in a part of the body that is, which is related to a statement given. And I'll give one example related to ownership and awareness, which is to color the body part that does not belong to me or does not exist. And one example for sensation and neuropathic pain, which could be color the body part that is painful and has altered sensation. These two tasks specifically are important to get feedback from adults with spinal cord injury. As you can see, the image is kind of just a placeholder, and I'd really love feedback from both PTs and specifically adults with spinal cord injury to give feedback on both the content of the items, of the sentences being asked, and also of the images that will get colored in. Next will be reliability and validity testing that I will not get into since there's a time constraint, but I'll be testing reliability in adults with spinal cord injury and without spinal cord injury, and then correlating these results with other known body awareness scales that exist. So reliable and valid MBR assessment is needed to identify MBR deficits in evaluation effects and to evaluate the effects of treatment and MBR outcomes in spinal cord injury. Here are my references and acknowledgements of funding. And I'd like to thank Dr. Ann Vandewinkle for her amazing mentorship and point out that Sarah Batale is the CMR therapist that conducted the initial item generation. Thank you so much.
All right, needed the posters for the question later. And if the next student, if you'd like a microphone, please go to the desk at the left-hand side of the room.
Okay, staying on. Oh, just use this one. Just use this one. You guys can hear me? Awesome. All right. So my name is Seth Darnell. I am an undergraduate researcher at Emory University in Dr. Floyd's lab. And today I'll be talking to you guys about sex as a biological variable in the immune response in the porcine model. So first, I wanted to give a little background on the inflammation and secondary spinal cord injury. So after SCI, there's two main phases, the primary and secondary injury. So mainly we'll be focused on the secondary injury and the acute and chronic phases of those. So we have a well-defined immune response and inflammation response of the rodent model in SCI. However, this is not very present in the porcine model, which is a higher translational model. So looking to characterize this will be very important as we work to develop further cures and knowledge in the injury field. And we also wanted to evaluate sex difference because we figured that might be an important factor in determining treatment plans and treatment methods. So kind of a brief overview of our methods of how we studied this. So we had male and female groups split into 3 different types of groups. So our control, laminectomy, and a contusion compression SCI model done at the T10. And so we split these into 2 time points, 2 day, our acute time point, and 6 weeks, our early chronic time point. And then we did ELISA, histology, immunology, SDS page, all these different laboratory methods to evaluate our inflammatory factors after injury. So now into our first set of results. So we looked at specific inflammatory factors, phospho-NF-kappa B and IL-6. So first for the phospho-NF-kappa B, we did a Western blot analysis where we looked at the laminectomy groups, male and female, pre and post injury. So we look at the SCI group on the right, and you see that there is a significant difference in a decrease of the female SCI group as compared to the previous one, compared to the laminectomy group, sorry. And then we look at the IL-6, we see a significant increase in IL-6, which is a common inflammatory factor in the females as compared to the uninjured group and the male group. Then we also looked at 5 different cell types. So we looked at neutrophils, microglia, macrophages, activated macrophages, B cells, and T cells. We evaluated these at both time points, the 2-day and 6-week time points. And we saw that neutrophil content significantly decreased as time went on, but in all other cells, there was an increase. And this helps us gain a better characterization of the immune response of the animal as time goes on after injury. And then we also evaluated the sex difference at 6 weeks post-injury. So we saw no significant change in all the cell types except for the T cells, which are a very common inflammatory cell and factor. So it was very significant. Once again, in the female as compared to the male, there was a much higher level of this. And so what we can draw away from this, we can observe that there is a significant difference in the immune response and inflammatory response between males and females after spinal cord injury. And this should be definitely considered as a significant factor going forward as we consider new treatment models and work on further experimentation as to consider sex as a variable. And is also very important to continue to characterize the immune response and inflammatory response of the porcine model as a translational model moving forward as we work towards a solution and just improvements. So that is all. Thank you very much.
Thank you so much. This is very enticing. Go out on a limb and make a wild guess. Nobody's going to quote you today. Is this possibly translating into altering hormonal interventions in the acute phase of spinal cord injury? And when you answer that, let's all us recall the discussion about the risk of acquiring SARS-CoV-2, aka COVID, as a male compared to a female. That speculation was rampant initially and subsequently sort of died down, and I still haven't heard the verdict as to what made males more susceptible to acquiring SARS-CoV-2. So, and that led to a whole discussion about giving hormones, attenuating hormones as therapeutic interventions. Do you see such interventions applicable in the acute phase after spinal cord injury?
Yes, I do believe that, I mean, granted, going out on a limb here, but I do believe that hormonal is playing into that difference we see. So I think those hormonal factors could be used as an acute response, possibly. Thank you very much.
Okay, hi everyone. My name is Valerie Dietz. I am currently a postdoctoral research fellow at Houston Methodist. And today I'll be talking to you about some of our transcutaneous stimulation work under Dr. Dmitry Sayenko, who gave a wonderful talk yesterday. So I'm going to just briefly go through some of our introduction. So spinal cord injury, as we know, is usually resulting from a discommunication between the brain and the rest of the body. So during injury, that connection gets interrupted and leads to a variety of symptoms, which we've heard a lot about here today and yesterday. However, some of these spinal circuits can still remain functional even after a spinal cord injury. And that's where neuromodulation can come in. So neuromodulation delivered in the form of electrical stimulation can be provided either epidural, which involves a surgical placement of an epidural paddle or a lead, or through transcutaneous spinal stimulation, which involves placing electrodes on top of the skin to deliver stimulation. And both of these modalities of stimulation have been shown to activate common neural targets within the spinal cord, resulting in similar levels of motor functional output. So with an epidural spinal cord stimulator, there's been a lot of interest in it over the past several years. And the first video that I'm showing on the left is showing what we're calling this enabling volitional control of task-specific muscle activity. And this video was taken in the first session of turning on the spinal cord stimulator. And that's one approach. And another approach by other groups in the field are using more of an inducing type of stimulation. So what I mean by this is they're inducing movements by precisely timing the stimulation pattern to get different specific motor pulls in this very programmatic fashion. And so this has all been done using epidural. Our research group wanted to ask, can we get these same effects using transcutaneous non-invasive spinal cord stimulation? And that's what I'll be showing here today. So in our lab, when we get participants, we first start by spending a lot of time doing TSS optimization. So we spend time figuring out where our participants will get the best electrode placement and what stimulation parameters give them the best motor functions for a specific motor task. And one of the ways that we're able to do this is we use a device called ExoLab. ExoLab is the picture in the middle, and this device measures force sensors in the ankle and knee. And we also use EMG over the lower limb muscles, so that way we can look at muscle function. So we pair these two devices together to get a real-time output of how stimulation is altering what force generation is coming for our participants. And across the bottom, these are a few examples of kind of how our methodology for moving through different stimulation arrangements. So we can do single stimulation from one electrode. We can do 2 electrodes at the same time. We can do 2 electrodes at the same time where we have the first one go off and then the second one, or we can also pattern them like what I mentioned before. So the first video I'm going to show you is of one of our participants. This participant has a T2 level of injury classified as Asia A, 4 years post-injury, currently 26 years old. And in this video, we're stimulating from the T11, T12 electrode. It's a continuous stimulation, and you're seeing some knee swings in this side lying position. On the right-hand side is kinematic data of us repeating this in a seated position. And you can see that we have lots of leg swings. There's a little bit of distribution in there, but that kind of goes into this volitional control that he's really thinking about making this knee swing. In this next video, this is more of our inducing effect. So we're getting this pattern stimulation where we have the red electrodes fire and then the green, and we alternate back and forth. And you can see in this video, we're getting a lot more extension through the end of that kick in this side lying position. And also again, on the right is our kinematic data from Vicon in our seated position. So that's 2 examples of how we're able to work in our transcutaneous stimulation to get this inducing and enabling effect. And so really the driving point here is that we were able to modify our stimulation parameters, modify our location of where we were providing stimulation to elicit the type of motor functions and outcomes that our participant was wanting to work on. And this work highlights the importance, and so did Dr. Saenko's talk yesterday, about really spending time with our individual participants, figuring out what stimulation works best for them in accordance with what motions we're trying to take them through. So just honing in on the fact that this optimization needs to be done in an individualized manner, and a one size does not fit all. So thank you to all of my lab members, to Dr. Sayenko, and of course to U2FP. This is one of my favorite conferences. I love the collaboration, and I would love to invite anyone who has questions to come find me at the poster session. Thank you so much.
Really, really nice work. From a day to day with the same subject, how does electrode placement, can you be consistent? And do you see the same kind of responses on a repeated measure from the same subjects? Because I think that's a pretty important way of demonstrating reliability and robustness in what you're doing.
Thank you for that great question. So yes, we see, so first of all, let me preface this. We see these participants through multiple weeks of training. So our goal is always to make sure that during these TSS optimization, that we are placing the electrode as close as we can to the same spot. And we check that with our EMG and our electrophysiology assessments. So before every session, we run like a really quick recruitment curve or something like that, just so that way we're kind of making sure we are in the same spot. And we also use like physical landmarks. We palpate the back of the spine to make sure that we're going in that area. And then, you know, like I said, we kind of fine tune it based on that recruitment curve. And we also, as kind of a fun trick that our physical therapist taught me, you can kind of like find landmarks on the skin. So like sometimes they'll have like a little mole or something, and that might also help, you know, make it a little easier to find that same spot. We have joked about that. We have not asked any participants to get a tattoo, but we've even said like maybe a henna tattoo or yeah, so I wish. Thank you guys so much.
I'm going to use the handheld microphone so I'm not gesticulating wildly. My name is Tucker Gillespie. I'm an undergraduate researcher in Dr. Jennifer Doolin's lab at Texas A&M University. So this talk is going to parallel well with hers. And I did want to give attention to all of the names on this slide because this was a big project, a lot of data, and we could not have done it with all of their help. So this project is looking into a systematic analysis of federal funding for SCI research. Obviously, we're all interested in both advocacy and research, and a large part of that is really seeing where the money is going and how the money is being used. So on this first slide here, this is just the federal funding broke down. And so we see funding from the NIH, the DoD, and the VA. And the large majority of this is still coming from the NIH. And so that is where we started. I did include the Craig Nielsen Foundation down below because there's a large amount of private funding that goes into this as well. We just were not able to include that in this portion of the study. So we started with the NIH, and then the question is, how do we access this data? Luckily, the NIH reporter allows us to access these public funds. However, we only have access to the PI generated abstract. So there's not a ton of data available. And as you can see, this is a slide of what happens when you search the keywords spinal cord injury, which is what we used in this study. We searched spinal cord injury, and then we downloaded all of the abstracts for the associated grants. But in order to understand what this money is being used for, you have to read and digest that abstract. There's no way to get sort of a objective bird's eye view and understand where this money is going and what type of research it's funding. So that was the purpose of this project. So we went through and had each line of data and each abstract was categorized by at least three individuals. And we added tags for each of these three categories. The system problem, really looking at what problem is being addressed, why is this study being done? The approach, how are we addressing this? Are we looking at mechanisms? Are we using a device? And then finally, the readiness. So asking, is this translation, basic research? Are we already progressed into the clinical phase? And this allows you to have a pretty good objective view of where this money is going. So I don't have a lot of time, but I did want to share some of our beginning approach intervention results. And this is only for the R grants. So only for the R grants. And this is just total dollars for each approach intervention. So as you can see, there's quite a few categories, but it sort of comes out to be a bimodal data shape. So up at the top, I like to call these our discovery group. They're really looking a lot into mechanisms or gain of function and loss of function. And in that group, we see the most amount of funding going into in vivo approaches. So any sort of animal that's not utilizing a official therapeutic intervention. And then in the middle here, we have more of a therapeutic intervention, and you see a fair amount of funding in all these categories, but especially into drug interventions and devices, which it sounds like we've heard a lot about this week anyway. And then we were also able to stratify this data by year. So when we pulled this data from the reporter, we only had up to 2022. And so we have data available from 2008 to 2022. And you're able to kind of see some of these trends over time in this heat map. And so again, with the in vivo approaches is taking up a lot of funding in that discovery category. And even that, you're seeing a pretty substantial increase in the last 4 or 5 years. And then here in some of the therapeutic interventions, I really thought it was interesting that you're kind of starting to see a little more isolation. Some might be getting slightly less funding, and we're increasing funding in areas like invasive devices. And then this also shows you some of the areas that aren't being funded and may remain untapped. These might be things that we have looked at in the past and have proven to be ineffective, or it might be new areas that research dollars could really benefit and could really be utilized and discover new therapies. So any questions?
So a couple questions. First, with the data, oh, can you hear me now? With the analysis, did you guys manually sort through the 1,000 studies, or did you have any type of machine learning or data programming done? I know Ingo is a database.
So this was all manual sorting. And so that's why I said each line was reviewed by at least 3 individuals. We had 4 main groups. And so each group did around 200 lines of data. And so we were individually reading these abstracts, and then we had descriptions. So I don't have my slides anymore. So those categories, we just included the names, but we had pretty detailed descriptions and parameters for what was included under invasive devices or so on and so forth. And so we were reading those abstracts and then gathering data from there and then manually categorizing them.
Is there plans like with the creation of a database to do some data programming to where it will automatically categorize, or?
Yes, ma'am. And I'm happy to talk about that more at my poster as well. But that is in our future plans.
No.
Yes.
No. No.
Good afternoon, everyone. My name is Jacob Goldsmith. I'm a postdoc at the James J. Peters VA Medical Center in the Bronx, New York. I work under Noam Harel and Jill Wecht. Today, my poster is about my protocol for my career development award project that I'm just started. But I wanted to use this presentation as a way to set up some background, and then hopefully people come talk to me on my poster. So as we know, spinal cord injury results in impairments in sensory motor function, corticospinal excitability. This leads to muscle weakness, paralysis, neural and autonomic dysfunction, and increased risk for both cardiometabolic, cardiovascular, and it's not here, but even neurodegenerative diseases. We also know after spinal cord injury, 45 to 60% of men exhibit low concentrations of testosterone, and this can exacerbate many of these deleterious effects of the injury. And so we know low concentrations of testosterone associated with numerous health impairments. You can see some listed here. And what do I mean when I say low testosterone concentrations? I'm talking about hypogonadism. And according to the Endocrine Society, that's anything less than 264 nanograms per deciliter. So according to the Endocrine Society, if you're between 264 and 916, you're at physiological levels and you should be functioning well. And below this, many body systems do not function well. And we also know there's an age-associated decline in testosterone. And here you can see across the age span, the gray bars are spinal cord injury and the black are non-injured controls. Across the age span, there is an increased percentage of low testosterone observations for both total and free testosterone. And so I'm using these terms like total, free, bioavailable testosterone, and I'll make this point a lot in my poster, but it's important to look at this entire profile. And most of the time what's looked at is just total testosterone, and it misses a lot of it because after injury, the binding proteins, albumin and sex hormone binding globulin, are also affected. So it's important to look at the whole profile. And this is showing how testosterone is produced. Gonadotropin signal, follicle stimulating hormone promotes spermatogenesis, luteinizing hormone promotes testosterone production. But in a male that's receiving exogenous testosterone replacement therapy, this is going to cause strong negative feedback. That's not only going to decrease natural testosterone production through decreasing luteinizing hormone, but it's also going to decrease follicle stimulating hormone, which could lead to infertility. And so I mention that because I'm working with an intranasal form of testosterone that's short acting and so far in the literature may maintain gonadotropin levels and also preserve fertility, or at least certain aspects of fertility. So in the literature, we know there's a lot of associations with low testosterone with erectile dysfunction, life dissatisfaction, body composition, and cardiometabolic risk. And from the few replacement studies that have been done in spinal cord injury, we know that it can improve many things: energy expenditure, strength, body comp, bone health, etc. And from other populations and animal models, we know that testosterone can have pretty incredible cardiovascular and neural benefits. So I'm proposing that restoring physiological concentrations of testosterone may improve neural health to promote better motor and cardiovascular function after spinal cord injury. And I mentioned I'm working with an intranasal form of testosterone. And so any form of testosterone replacement has drawbacks and side effects. Some of them are listed here. But this intranasal form might be able to get around some of these. As I said, it's short acting, so it doesn't result in a strong chronic negative feedback in that system. It allows for peaks and troughs throughout one day, so no strong negative feedback, may allow fertility to remain. And it's also fast acting, can get to the central nervous system within 15 to 30 minutes, causing neural effects. And so it's suitable for studying these rapid effects. And so we know spinal cord injury results in cardiovascular and neural dysfunction. Low testosterone also exacerbates this neural and cardiovascular dysfunction. And we know TRT has numerous benefits, but there is a lack of information on its effect on central nervous system excitability and other neurophysiological parameters, as well as cardiovascular autonomic function. Only one study has looked at cardiovascular autonomic function in spinal cord injury population with testosterone replacement. And also this intranasal delivery may allow to preserve fertility and also allow us to study the rapid effects of this. And so primary aim is to look at the associations between the testosterone profile, central nervous system excitability, and cardiovagal reflex function. And then exploratory aim is to look at the acute effects of a single dose of intranasal testosterone on CNS excitability, cardiovagal reflex function. And then also we're going to do a 1 month follow-up to see how 1 month of sustained intranasal TRT results in adaptations. Thank you so much.
Great work. Thank you. 2 brief questions, please. Number 1, in the acute phase, aren't we supposed to think that the decrease in testosterone may have an adaptive role? Remembering that studying young recruits, Hell Week, and all of the other, we're studying 17-year-olds who are brimming with testosterone at basal levels, whose testosterone levels are undetectable during the stress of those weeks. Number one. Number two, in the chronic phase, do we worry about thromboembolic risks of testosterone supplementation?
Fantastic question. To the second question, I would say I have to be very careful to say physiological levels of testosterone. So I'm just talking about replacement levels. I'm not talking about super physiological levels. At that point, I think you would have to worry about those effects. And I'm so sorry, the first question. Oh, early on in the acute, based on what I've read, it could be. But what's interesting about that is it persists in 45 to 60% of men into the chronic phase. And that's what I'm working with the chronic population. Thank you.
Okay. Oh, great. Okay. So my name's Loudon. I'm a PhD researcher at University of Florida. So I just came here with Hurricane Helen. So missed yesterday because of that. So sorry about that. And although I'm a PhD, researcher, I am actually not here to talk about my research there. I'm talking about my startup, which is Intimistim. And this is going to be the most memorable U2FP presentation. I guarantee that. And I found out actually you can trademark something if you say it enough. So if we say it enough, it'll become trademarked. So great. So what we're trying to build is a minimally invasive neurostimulator to restore sexual function in the spinal cord injury population. There's 300,000, this is a little bit more for general audience, 300,000 SCI population in the US, and sexual function is actually the #1 request for, many in the population. Most are male, and there's not really any real solutions. Everybody's focused on walking. So a lack of sexual function causes problems for intimacy, love, and fertility. SCI patients are excluded from living a normal life. So the solution is to make a minimally invasive neurostimulator. So only the electrode is implanted. Everything else is worn externally. And so battery and computer are worn externally. And then the external portion are worn for about 4 weeks at a time, kind of like a continuous glucose monitor. And this is built on existing technology. So it should be a really fast surgical implant, nothing too new. And for the market, 300,000 SCI population in the US. If we go for only the early adopters and the innovators, we can have a nice revenue, but we can also expand this potentially to the rest of anybody who wants a sexual function boost, and it'll be much larger revenue. So we have gotten a good amount of traction. We've gotten some funding. We've won the Blueprint MedTech seedling grant, which is a $50K grant. And it was back when we were doing FemiStim. We've also won a Luby grant, local, grant at University of Florida and in Northern Florida, and another similar one too. And so the way we're planning to do this is at cost minimally invasive implantation. So because it is minimally invasive, it can be cheap. This is going to be expected to be paid out of pocket. Insurance doesn't really care about if you have intimacy in your relationship or not. So we want to bring this down as much as possible, basically provide it at cost. But the real revenue driver would be a monthly subscription for the wearable component. And we would expect a larger profit margin for this. And then a nice thing that we could be able to do that I don't think has really been done in medical devices is we could have Intimilink. So you could have simultaneous orgasm with you and your partner every single time by pairing devices. So hopefully this is exciting for anybody with SCI who would like to become a sex god. Yeah, big guys, sorry, big guys aren't interested in doing anything. And our team is not just me. We have a founder of another neurotech company who's been in it for over a decade, implantable neurosurgeon, and unfortunately Dennis Borbeau, he's a researcher, he couldn't be here. So yeah, we're submitting for a patent now, looking for more funding, non-dilutive, and always looking for lived experience interviews. I have a microphone and a recorder, so happy to do that. And if you have any special skills, then let's talk. Oh, any questions? Okay.
I have a question,
Yep.
LaDon.
Yep.
You know, yeah, I think you might know my question, but I'm just going to ask it anyway. Mechanical pleasure. Any plans, any thoughts, any ideas about how you can pursue pleasure side of the equation?
Yeah, so this is basically to recover erection ejaculation, but actually Elizabeth Bodorf's research is based on this, that you can potentially cover orgasm function as well. And in our research, and not asking our wives, apparently women have orgasms too, and so we believe this could work for women as well for a pleasure component as well. So it's a possibility. I don't want to promise anything though, but we can dream. We'll make it happen. So thank you.
All right, hi everybody. I am Tommy Suter. I'm another Gator, and I'm gonna jump right into telling you about assessing the significance of individual breathing responses to neuromodulation interventions. So right off the bat, I wanna thank my postdoctoral mentors, Dr. Emily Fox and Dr. Gordon Mitchell. My co-authors on this project are funding from the Department of Defense and our participants who make all of this possible. So at my poster, I'm going to be using data from the BioFlow clinical trial, which is currently ongoing at Brooks Rehabilitation in Jacksonville, Florida. In the BioFlow clinical trial, we're assessing acute intermittent hypoxia, which you heard about yesterday from Gordon Mitchell, as well as acute intermittent hypercapnic hypoxia, which is another fluctuating gas mixture against a sham placebo control. One of the outcomes that we're assessing is minute ventilation, which is quite simply how much air you breathe when you're at rest. So we're looking at that before and after each of these three interventions. We have data so far from 5 participants who have completed the entire trial. Now, if you analyze their results for minute ventilation in a traditional group analysis way, what we actually find is that there isn't a significant effect of either intervention. So what does that tell us about the individual effects on each person? At my poster, what I'm actually going to be talking about is exactly that, the significant effects that each individual experienced. And I'll be telling you about how 2 of them did not experience a significant change in their breathing, but 3 of them did. So the exact methodological way that I'm doing that, I'm going to save those details for my poster because I want to use my slides as an opportunity to talk about why would we look at the data this way. Thankfully, over the last 2 days, everyone else has already talked about participant variability and response variability. We've all heard that no 2 spinal cord injuries are the same. Everyone presents with different baseline function and ability. Additionally, everyone responds much differently, even to the same interventions. But going beyond looking at participants like this, participant variability and response variability can also refer to the same one person. We've already seen over the last 2 days how there are some individuals who have responded differently to the same intervention when measured at different time points. People have more or less energy, they feel stronger or weaker on different days, and so those things can also influence how they're going to respond to an intervention and what their baseline is from day to day. So this is important when we want to characterize responders to interventions, which we've heard already there's a number of different ways of doing. And I believe that the individual statistics are one way of characterizing those responders. For instance, in the BioFlow clinical trial, the bio part of that name refers to biomarkers that we're looking at. We're looking at the presence or the absence of specific genes that people who do and don't appear to respond to intermittent hypoxia have in common. I think that using these individual analyses in conjunction with things like biomarkers are going to enhance understanding of how we lead towards personalized treatments for every individual person with a spinal cord injury. So at my poster, I don't claim to have this, solution that's going to tear down research and rebuild it from the ground up. I want to start a discussion. Doing group analyses is important. We need to understand how a new intervention generalizes to the population. We want to know about biomarkers so that we can classify people that do and don't respond. And the real world significance beyond the statistics of whether an intervention helps you live your life better is probably the most important thing. What I'm proposing is that we use these individual analyses as another tool in the box to lead towards those personalized treatments and ultimately benefit the whole spinal cord injury community. Thanks.